Okay, I think we're gonna start. So good afternoon and welcome to the second half of our symposium. We have started recording, we have unmuted, so I think we're ready to go. So I want to introduce our next speaker, Dr. Radhika Gogoi. Radhika is a physician scientist and gynecologist. She is an associate professor in the Department of Oncology and OBGYN here at Wayne State, with a clinical practice at Ovarian Cancer Center. Her research focus is in the early events leading to initiation of ovarian cancer, particularly the role of HIPPO pathway and its impact on EMT. Radhika.
Yeah. Great. So thank you, everybody. My name is Radhika Gogoi, as Aisha mentioned. Welcome back after lunch. Hopefully everybody ate and not too much, so our metabolism is nicely revved up. So my talk today is on connective tissue growth factor, or CTGF, which is as a marker of EMT. And the picture really sort of describes hopefully what my data shows and really my approach to this talk. So with that, I'll get started.
What happened for this to be now? What's the movie?
I'll give you the ending at the end.
You have to click on the. Yeah.
Okay, perfect. Yeah, I think so. So I'm going to start with the last slide first, because this is really the most important slide. And if you all take a nap after that, I won't be offended. But I wanted to just make sure I give appropriate thanks really to Dr. Moore for his leadership and for really taking me in and adopting me into this wonderful organization, which is the Mott Center. Dr. Alvaro for her mentorship. Sandra, who is really the key to all of this. There should be a halo instead of the star, because she certainly deserves it. She's done all the work on the project that we're going to talk about. The Division of GYN Oncology, my partners, Dr. Roblick, Dr. Winer, Dr. Morris, my fellows who are here today. Of course, the Mott Center for Human Growth and Development, and most importantly, our patients and our families who fight courageously really to overcome this disease. The other person I really need to thank, and a person that I wasn't really very familiar with, is this young developmental biologist. So her name is Betty Hay. She's really thanked for the development and models of EMT that we will talk about. She's an American cell and developmental biologist. She is first established the concept of epithelial to mesenchymal transition, really in the process of gastrulation. She demonstrated EMT in 2D collagen cultures and also demonstrated the role of ECM in driving cell behavior. And she is really who we have to thank for sort of the process of EMT. So when we talk about EMT, we're really talking about the transition from an epithelial to a mesenchymal transition. This occurs during both normal development and in pathology, and it's often incomplete. So the process often results in intermediate states, which are sort of the epithelial states between epithelial and mesenchymal cells that retain both epithelial and mesenchymal properties. In 2020, there was a consensus conference that came together to really define some level setting for guidelines and definitions for the research on epithelial mesenchymal transition, and adopted sort of the term epithelial mesenchymal plasticity, which is really the preferred term to describe the ability of the cells to adopt mixed epithelial mesenchymal features. And they also suggested that researchers should describe both epithelial mesenchymal transition as well as mesenchymal to epithelial transition as functional changes in the biologic properties of cells rather than on specific molecular markers. Now, on the right here shown are sort of what they describe as core EMT changes, and you'll see why these are important. They include cytoskeleton remodeling, loss of apical to basal cell polarity, cell-to-cell adhesion weakening, cell-to-matrix adhesion remodeling, cell individualization, acquisition of cell motility, and basement membrane invasion. Sort of these are the core changes that are necessary for the process of EMT. So why is EMT important in ovarian cancer? And some of this data really came from the 2011 TCGA integrated analysis of high-grade serous ovarian cancer. And you've heard some of the other speakers as well talk a little bit about this analysis. So the TCGA was the largest data set at the time in 2011, utilized 489 samples of high-grade serous ovarian cancer, a fairly significant follow-up period of 60 months. And when they performed the TCGA or exome sequencing analysis, they really identified 4 groups based on specific gene signatures. And those 4 groups were really the differentiated group, immunoreactive, mesenchymal, and proliferative. And of course, this is interesting because it is really the mesenchymal group of genes and the tumors that have those genes that are really interesting to us. So what does this mean about what the outcome is in these 4 different groups? And so in 2014, a group looked at the TCGA cohort, which we just spoke about, and a separate group from a separate cohort from the Mayo Clinic. And they really established sort of what the overall survival was in tumors that fit those 4 signatures. And so what you'll see here is that the immunoreactive group, really the top line in black, had the best survival. And the mesenchymal group shown here in the red really had the worst survival. So mesenchymal features certainly seem to play an important role in that specific cohort. To validate their findings, they also did the same thing in the Mayo Clinic cohort. And again, really found that the immunoreactive group shown in the black really did the best. And again, the mesenchymal group here shown in the red had really the worst overall survival. On the right are really sort of a breakdown of those features. What you'll see is that the mesenchymal tumors with the mesenchymal phenotype had the lowest R0 resection rate and overall the worst prognosis. They were characterized by sort of what you saw already were the core features of EMT, including focal adhesion, ECM receptor interaction, activation of the JAK-STAT signaling pathway, TGF-beta signaling, and VEGF signaling. So we know that not only are ovarian cancer tumors heterogeneous, but tumors that sort of specifically show the mesenchymal phenotype do in general worse. So you've seen, it's nice because some of this stuff already has been shown by a number of other speakers. And so we know that in epithelial ovarian cancer that it's a process. And so the earliest, as Dr. Matei presented, that the idea is that the fallopian tube is really the origin or site of origin of high-grade serous ovarian cancer. And shown here is a schematic with normal fallopian tube epithelial cells, the conversion of those into serous tubal interepithelial lesions, stick lesions, which are have a p53 signature, and then specifically the initiation of high-grade serous ovarian cancer. So a lot of my talk on connective tissue growth factor really starts in this very early transition to EMT and not sort of some of those late effects of EMT, which are shown in the next slide. Again, normal fallopian tube, the initiation of a p53 signature leading to high-grade serous ovarian cancer, and then specifically the other features of a mesenchymal cell, which include invasion, metastasis, and then basically invasion through the basement membrane. So just wanted to point out specifically sort of the features on immunohistochemical analysis of both stick and still lesions. So shown here on the right is the still lesion, which is a serous intraepithelial lesion. It's characterized really by a p53 signature and really a low Ki67 signature. On the right are the stick lesions, which are believed to be precursor lesions to high-grade serous ovarian cancer. You can see the difference in that these really have a much more, the cells are piled on top of the other, they lose apical basal polarity, they have a fairly significant p53 signature, as well as a MIB1 signature. And again, it's believed that these are really the precursors to the high-grade serous ovarian cancers. So as you all know, and has been nicely pointed out by all the speakers here, the problem really is that advanced or metastatic ovarian cancer is associated with a decrease in survival from almost 95 to 35%. And so the identification of the metastatic triggers of EMT, their targets, and what controls the process are really the unmet need. And so the goal of the research in the lab is really to understand the factors and targets that are critical to the transition between the epithelial and the mesenchymal phenotypes. And so if we understand what allows the transition to EMT, perhaps we can find targets that are then therapeutic. And so the hypothesis for the lab is really that the connective tissue, this protein called connective tissue growth factor, is critical to maintain the epithelial phenotype in ovarian cancer, and that the loss of CTGF is one of the early markers of EMT. So what is CTGF? CTGF is a protein that's both produced in the cell as well as secreted. It belongs to a family of extracellular matrix-associated heparin-binding proteins, and CCN2 is really CTGF. A knockout of CTGF dies at birth. This is a mouse model with severe chondrodysplasia, and conditional knockouts in the uterus and the ovary are associated with subfertility and disrupted follicles. CTGF, my specific interest in CTGF was because of the Hippo pathway, and really it's been shown to be a downstream target of the YES-associated kinase or YAP, and the Hippo pathway. So on the right is a schematic of sort of CTGF activity. As I mentioned, CTGF is produced both in the nucleus and is secreted. It's associated with a number of other physiologic processes, pathologic and physiologic, including angiogenesis, ECM stiffness, gene expression programming, cell motility, proliferation, adhesion, cytoskeleton remodeling. It also, as a cytokine, binds to a number of other receptors, including VEGF, EGFR, and leads to transduction of STAT3 pathway. So a good target really for what we consider as processes associated with EMT. In cancer, CTGF has been shown has sort of a dual role in that there are papers that show that overexpression of CTGF is both contributes to the development of cancer, as well as papers that show that a loss of CTGF is associated with cancer. And that's true in high-grade serous ovarian cancer, as well as in other cancers, including breast cancer and gastric cancer. So the take-home for CTGF's role in cancer is that it really seems to be both tumor type dependent as well as site dependent, in that its expression may be dependent both on the stage of the tumor as well as the type of tumor that it's involved in. This is a cartoon just showing sort of the promoter of CTGF, and it's again regulated by multiple pathways, specifically TGF-beta activates CTGF, as well as YAP, which is the protein associated with the Hippo pathway. It can bind to multiple receptors, including integrins, heparan sulfate proteoglycans, lipoprotein receptors, tyrosine kinase receptors. And not only can it bind to it, but it can bind specifically to VEGF, EGF, BMP4 as well, and can bind to a number of extracellular matrix proteins, including fibronectin and aggregatins. So our lab studies models of EMT, and this is a cartoon showing a model that we use in the lab that you'll see the data really utilizes this specific model. So we have primary epithelial ovarian cancer cells. They're called the R182 cell lines that undergo, during the process of EMT, undergo really loss of apical basal polarity, a change in the structure of the cell, as well as the ability to invade. And this epithelial mesenchymal plasticity leads to the mesenchymal cell line, which is the MR182 cell. And Dr. Teja is nicely, who I borrowed this graphic from, shows really the epithelial cells, which you can see are normal epithelial cells, and their transition to this hybrid state with really the loss of the structure, and then the mesenchymal state, which allows spheroid development. So the R182 and the MR182 cell lines are the ones that we'll use for the majority of the cell models that we'll talk about. So to understand what are the triggering factors of EMT, Gil did a RNA sequencing analysis looking at differentially regulated genes between the R182 epithelial cells and the MR182 mesenchymal cells. And so that would be to try and understand what are the processes and the genes that are associated with the process of epithelial mesenchymal transition. And lucky for me, the hippo signaling pathway came up as one of the top 3 differentially regulated genes. And again, when we look specifically at the differentially regulated genes, shown here is the volcano plot. Blue are downregulated genes, red are upregulated genes. CTGF comes up as a differentially regulated gene that is lost in the transition between epithelial and mesenchymal transition. On the right is the biological processes that are associated with EMT, and as you would expect, a lot of these are extracellular matrix remodeling, including actin cytoskeletal reorganization, integrin activation, and epithelial cell-to-cell adhesion. So given our interest in the hippo pathway and in CTGF, we wanted to see first really screen a number of ovarian cancer cell lines that we had in the lab to see what the level of expression of CTGF was in these cell lines. And so we screened epithelial as well as mesenchymal cell lines for CTGF. And what we sort of noticed was that epithelial cells were more likely, had a higher expression of CTGF than mesenchymal cells, which seemed to lose CTGF expression as they became mesenchymal. So to understand what CTGF did, Sandra knocked out CTGF using a CRISPR-Cas9 plasmid in both the cell lines. We have 2 epithelial cell lines, and really screened them for expression of both epithelial and mesenchymal markers. So on the right is a Western blot looking at the epithelial cell line, our knockout cell line, and then the mesenchymal cell line. And again, in CTGF, you can see that we lost CTGF as we expected, and that the mesenchymal cells don't express CTGF. Interestingly, the knockout cell continues to express the epithelial marker beta-catenin as well as CK18, doesn't express the classical mesenchymal marker twist, which is expressed in the mesenchymal cell line, but interestingly does express an earlier mesenchymal marker called snail, which you can see is present in the knockout cell line. So taken together, this data suggests that the CTGF knockout cell line really represents sort of that epithelial mesenchymal plasticity, or an intermediate state between an epithelial and a fully mesenchymal cell line. So we wanted to look at what the effect was on characteristics of mesenchymal cell, and one of those characteristics is really anoikos resistance. And anoikos resistance is the ability of a cell to attach in the absence of a substrate or a extracellular matrix. And that's really a characteristic of a mesenchymal cell. So we took the wild type epithelial cells and the knockout cell line, plated them in an ultra low attachment plate, and looked to see whether these cells would survive in that substrate or in the absence of a substrate. And what you'll see here, both by picture and by quantitation, is really that the wild type cell loses its ability to attach by 48 hours. So there are no viable cells left. In contrast, when you lose CTGF, at 24 hours, there's still a number of cells that are able to form clusters and survive. And at 48 hours, that's still true. So the loss of CTGF permits anoikos resistance. And again, shown here, the similar finding quantitated, where you can see that the wild type cells are not viable by 72 hours, while the loss of CTGF permits cell viability. The second thing we wanted to look at was the ability of cells to invade when CTGF is lost. And so shown here, these cells were plated on 50% Matrigel, and invasion measured at 160 hours. And you can see that the wild type cells really don't invade. Our epithelial cell line does not invade. The CTGF knockout cell is able to invade at 160 hours, and that when you add recombinant CTGF back into these cells, it really resembles that of the wild type. And that's shown here in the graph as well, or in the pictures. So once we found phenotypes that were associated with loss of CTGF, we wanted to see really the mechanism of action of CTGF. What is it that CTGF loss is allowing it to do? And so we did, again, an I-pathway or an RNA sequencing analysis, comparing the wild type R182 cells to those that had the knockout of CTGF. On the left are the pathways that are differentially expressed between the knockout and the wild type. And you can see that really, as you would expect with understanding what CTGF does, is that cell adhesion molecules are a significantly differentially regulated pathway, as well as ECM receptor interaction, consistent sort of with its role as an extracellular matrix protein. The biologic processes, again, extracellular matrix organization shows up as a significantly regulated biologic process, as well as cell adhesion. So we wanted to explore that a little bit further. And one of the proteins that shows up as being differentially regulated when we look at the genes is LAMC2. So this is just a slide sort of introducing laminin C2. It's a gene that encodes the gamma subunit of LAM332. And what it is a large extracellular matrix glycoprotein that's found exclusively in the basement membrane. And that's the important part, is that this is a protein that loss of CTGF upregulates that is only found in the basement membrane. It is implicated in the autosomal recessive disease epidermolysis bullosa, and it's a biomarker in a number of different cancers. And so on the right is just a cartoon of sort of what a basement membrane is. Again, this is the epithelial cell. And the basement membrane really consists of sort of muscle cells, adipose cells, endothelial cells, Schwann cells, for example, in the neurons. And really the main protein associated with the basement membrane is laminin C2. So our interest in this really came from this data, which is, again, RNA sequencing analysis looking at differentially expressed genes between CTGF knockout and wild type cells. And we were interested in LAMC2 because, again, it was statistically significantly increased when you lose CTGF. And this is shown here on the right as well by PCR. We validated that loss of CTGF upregulated LAMC2. So you wanted to see what happened on a protein level when CTGF was lost. On the right is the Western blot looking at the wild type. And what you'll see is really that LAMC2 is overexpressed, both at an RNA level as well as a protein level when you lose CTGF. And so one of the interesting things about LAMC2 is not only is it produced by the cell, but it's also secreted. And so Sandra looked at the supernatant in these cells. And again, what you'll see is that both the unprocessed, that's the higher band, as well as the processed LAMC2 are increased in the loss of, in the CTGF knockout cells. To confirm our results, we looked at immunofluorescence in the cell lines. The green is the LAMC2. The top row is the R182 wild type, which has small amount of LAMC2. And what you'll see, again, confirmed by immunofluorescence, is that loss of CTGF increases LAMC2. And when you add recombinant CTGF back in, the levels of LAMC2 sort of are similar to what was there in the wild type. So similar to the theme, we show again that reorganization of the extracellular matrix is really a key feature associated with loss of CTGF. And so to study that, I think you've seen, Dr. Alvaro also showed some immunofluorescence. So in green, this is a phalloidin stain, which stands for F-actin, and then the blue is the DAPI, and then this is the overlay. So the top is the R182 cells, and what you'll see is that, they have a nice actin cytoskeleton that sort of forms the rim of the cell as you would expect in an epithelial cell. When you lose CTGF, you can see that there's a disorganization of the actin filament. You can see these little filopodia that sort of extend out from the cell. And what you'll see here really is a sort of a disorganization of the extracellular matrix, and that's shown better in the enhanced pictures here. This is the wild type, which shows sort of a nice scaffolding of the apical of the cell by the actin, and in the lot with the CTGF knockout, you sort of see this disorganization or haphazard expression of the actin filaments. We also wanted to see really what does this do to the capacity of tumor formation, or what is the tumorigenic potential of CTGF knockout cells. So we took NSG mice, treated and injected them sub-Q with either CTGF knockout or wild type R182 cells. And we looked for tumor burden. And again, this was a sub-Q model. And what you'll see is that really the wild type, as we have seen before in our models, Wild type R182 cells rarely are able to maintain tumor, but the CTGF knockout cells were able to maintain tumorigenic capacity. So based on the data that I've shown you, and now we've just recently published, this is sort of the proposed model, which is that in the wild type or normal epithelial cell, for example, in the fallopian tube, we would hypothesize CTGF is expressed, allowing the cell to maintain really apical basal polarity, a normal actin cytoskeleton, a normal relationship with the basement membrane. Upon loss of CTGF, these cells lose some apical basal polarity. They have actin filament disorganization. They have a sort of dysregulated relationship with the basement membrane with increase in integrins. I showed you the increase in LAMC2. They also acquire anoicus resistance, the ability to migrate, and now the ability to form or maintain tumors. So we hypothesize that the regulation of CTGF, or we conclude that the regulation of CTGF and its downstream signaling pathways are critical to maintain the epithelial phenotype, and that loss of CTGF is one of the very early stages necessary for epithelial mesenchymal transition, and that occurs through remodeling of the extracellular matrix. So this led us to more and more questions, and this is really where I think all of you are great collaborators and have a whole lot of expertise in these areas. So there are now a number of different questions that come up based on that hypothesis. So if CTGF loss is an early event in EMT, are the changes really occurring then in the fallopian tube? Because we think that that's the site of origin of high-grade serous ovarian cancers. Other questions, what are the factors in the tumor microenvironment, if that's really where it's coming from, that regulate CTGF expression? And I'll show you some preliminary data about that. What are the downstream targets of CTGF? I've shown you LAMC2, but there certainly are other targets of CTGF downstream. And then, of course, the end result is what are the therapeutic targets really that would allow us to maintain CTGF expression and prevent really the transition from an epithelial to a mesenchymal phenotype. So this is preliminary data that hasn't been published yet that we are sort of in the process of looking into further. So the first question really was, if CTGF loss is an early event in EMT, are these changes then occurring in the fallopian tube? And so these are immunohistochemical stains. And again, these are a very small sample. We certainly need more collaborators and we need more samples to really to make a definitive conclusion about the expression of CTGF in fallopian tube epithelium. So these are normal fallopian tube. This is a stick lesion. This is high-grade serous ovarian cancer. And we stained for CTGF, snail, and beta-catenin. And what we would like to see is that obviously the normal fallopian tube epithelium does express CTGF, that there's a persistence of CTGF expression in stick lesions, but that there are some cells here, and again, it doesn't project great, that are layered on top of the other that seem to lose some CTGF expression, and that high-grade serous ovarian cancer really does not express much CTGF. Again, work in progress. Obviously one stick lesion doesn't make the statement well enough. So really what we'd like to do is really study this both more in a mouse model, if that's possible, or in patient samples to really try and understand where in the fallopian tube epithelium this is really occurring. So the second question we had asked are, what are the factors in the tumor microenvironment that regulate CTGF expression? And so iPathway Guide, when you look at the RNA sequencing data, has a prediction model that predicts upstream predictors by iPathway. And interestingly, what iPathway predicted is that miR-199, or microRNA, which I was really excited that you had already sort of set that up for me, Mr. DeFeo, was upregulated in CTGF knockout cells. And so, as you already heard from Dr. DeFeo, microRNAs are small, single non-coding RNAs that regulate gene expression. Most microRNAs are in fact involved in RNA silencing. And again, here what you'll see is in a typical gene, messenger RNA is produced that directs protein assembly and subsequent protein formation. The presence of a microRNA binds to the messenger RNA and ultimately, for the most part, blocks protein assembly. So we wanted to look at what whether we could validate the I-pathway guide results for miR-199 and miR-214. And so Sandra looked at PCR for both R182 cells as well as the CTGF knockout. And we find that, in fact, miR-199 and miR-214 do go up when CTGF is lost. So to confirm that, to see really what the effect of miR-199 is on CTGF, Sandra did a Western blot that showed, again, in R182 cells, there is some CTGF, and same with the negative control. But when you transfect in miR-199, you lose CTGF, suggesting that the presence of miR-199, in fact, binds CTGF and allows for loss of protein expression. So the next question we have is, where is this miR-199 coming from? And again, hypothesis yet to be confirmed on a larger scale. But one of the hypotheses that that the lab has is really that this is coming from the adipocyte. And so we wanted to look at, this is data that Dr. Teja has, looking at the expression of miR-199 in omental extracts. And so in collaboration with colleagues at Henry Ford, they took omental tissues, collected exosomes, and looked at miR-199 expression by PCR. And so what you'll find specifically is that obviously not all omental derived exosomes contain miR-199, but miR-199 is expressed in a certain percentage of omental exosomes. So that would be the hypothesis. So the next question is really, what are the downstream targets? So once miR-199, once CTGF is lost, LAMC2 goes up, but are there other downstream targets? And so because of its role as a cytokine, we were interested in IL-6 specifically, expression when CTGF is lost. And so this is just a slide that goes over a little bit of what we know about IL-6 expression in ovarian cancer. As you all probably are better aware, even more than me, it's a pro-inflammatory cytokine present in the tumor microenvironment. It's produced by a number of cell types, including neutrophils, monocytes, fibroblasts, endothelial cells, and tumor cells. It can be produced in response to TNF-alpha. It functions as a number of roles, specifically both in recruiting neutrophils and then cancer promotion, specifically migration, growth, activation of T lymphocytes, and stimulatory effects on cancer cells. So in ovarian cancer, it was found to be found in acidic fluid, that the IL-6 levels were prognostic of PFS, and that high serum levels of IL-6 have been associated with aggressive metastatic ovarian cancer. So it's interesting to us also because IL-6 is known to have a role in EMT. It is an important trigger of EMT. So IL-6 is the ligand that binds to the IL-6 receptor and then subsequently activates STAT3 and a number of other pathways, both associated with signaling, resistance to chemotherapy, MMP expression, loss of E-coherent, promotion of the cell cycle, and really allows the transition to a mesenchymal phenotype. Interestingly, an IL-6 antibody was tried as a phase 2 single-arm study in platinum-resistant high-grade serous ovarian cancer. In that group, specifically 18 patients tolerated it well, but unfortunately, they didn't show any response as a single agent. So given that CTGF increases IL-6, we were really interested to see what we know about levels of CTGF in the knockout cells. And so this is an ELISA that measures IL-6 levels in the media of CTGF knockout cells. And what you'll see is that there is a huge increase in IL-6 secretion in CTGF knockout cells, both in the R182 epithelial cells as well as the other cell line. Again, suggesting that loss of CTGF increases IL-6. We wanted to look at expression level of the receptor now that we know that the ligand is present. And this is done by immunofluorescence. The green again shows IL-6 receptor. And what you'll see in the wild type cells is that there is certainly IL-6 receptor expression, but a higher level of IL-6 receptor when CTGF is lost. So we've shown that there is the ligand is increased. We've shown that the receptor is increased, and then we wanted to understand what is if the pathway, specifically the classical signaling pathway for IL-6 receptor, is activated. And so to do that, we did a Western blot that looked at the wild type and the knockout cells, and we looked at downstream targets of IL-6 receptor binding, which is really phospho-STAT3. And so shown here on the top is the R182 cells, and you see phospho-STAT3 really turns on within 25 minutes and goes up to 1 hour and then turns off. And interestingly, that level of activation is different when CTGF is lost, notably that the phospho-STAT3 level continues to stay up till about 4 to 6 hours. What's also interesting to us was, and unfortunately, we were hoping that phospho-AKT would have shown up as being differentially regulated, but at least on a Western level, the level of phospho-AKT doesn't seem to change whether CTGF is present or not. So to conclude, we concluded that EMT is a tightly regulated process, or this is well known, we didn't conclude that, but which is essential to the process of metastasis, that CTGF expression is important in maintaining the epithelial phenotype, and its loss is one of the early markers of mesenchymal transition that we believe is due to ECM reorganization and remodeling. We show that miR-199 regulates expression of CTGF, and that the loss of CTGF increases ECM reorganization, both by increasing LAMP2 and increased expression of IL-6. So really, future directions that we'd really like to take this project is really to understand the factors in the tumor microenvironment that produce miR-199 and that regulate CTGF expression, to evaluate other downstream targets and the effects of both IL-6 and IL-6 receptor activation, including specifically my interest in the immune phenotype, to study how the loss of CTGF affects reorganization of the extracellular matrix, and then to evaluate therapeutic approaches that prevent EMT by trying to maintain expression of CTGF.
Thank you for sharing your data. Does the CTGF, is it responsive to like TGF beta or MIR-200 that are known to be regulators of EMT?
Yeah, it's interesting. So it definitely is responsive to TGF beta. So we've treated with TGF beta and we've seen CTGF expression. MIR-200, I haven't seen. It's probably in the upstream regulator predictor, but I have to take a closer look. miR-199 is the one that came up sort of quickly.
Great work. So I see another microRNA talk. So it looked like in your tumor genicity in vivo studies, it looked like the tumors just stayed static, right?
Yeah.
So did you do any IHC on those tumors to confirm that they were, like, what subtype? Were they high-grade serous tumors?
Yeah, so that's a great question. So yes, interestingly, the presence of CTGF alone doesn't seem to make them grow or invade, which is what we would have hoped. So certainly it may play a role, but it's not the only role. And it seems that it would require more than just loss of CTGF to sort of become fully mesenchymal in that it doesn't proliferate.
I'm wondering, like, if you looked at any EMT markers in those little tumors that grew, and maybe it has to do with the immune component, right? Because you just said that was in NSG mice, right?
Yes.
I wonder if you were to do it in nude mice that still have some immune, right? They still have NK cells. Would you get maybe some type of tumor growth, or if you were to add IL-6?
Yeah, so that's a great question. We did IHC. We definitely see tumor cells. They look like the high-grade serous ovarian cancer cells. We haven't done the immune cell by IHC, so that would be a great question. But certainly, this seems to require more than the loss of CTGF to maintain and to grow or invade.
Yeah. And one other thing, so there's actually 2 microRNA 181 sites in CTGF.
Yes.
And that in our models, we actually see dramatic loss
Wait, it was microRNA 199 or
181.
182? Interesting, there's also in the 3' UT HERF CTGF, there's a 199 binding site.
Yeah, and actually in our models where we get tumors to grow in the fallopian tube models, CTGF goes down dramatically throughout the process. So if you wanna,
Oh, then, yes, yes,
I'll be
absolutely.
happy to share that with you.
That would be wonderful. That's
Yeah.
exactly what we're looking for. That would be perfect.
Was not on the video?
Now it is.
Now it is.
Thank you, Radhika.
All right, moving on. I want to introduce our next speaker, Dr. Ron Chandler. Ron is Associate Professor in the Department of Obstetrics, Gynecology, and Reproductive Biology at Michigan State University, where he is also the Associate Director of the Cellular and Molecular Biology program. Ron's research focus is in the mechanisms involved in chromatin remodeling, steroid hormone signaling in the endometrium, and how disruptions in these contribute to malignancies such as Endometriosis-associated ovarian cancer. Welcome, Ron.
Thank you. Thank you. I'm gonna see if I can position this better. I can actually use it.
You have also the mouse there.
I think I'm good. So we're gonna, first I would like to thank the organizers, including Gil and Aisha, for a great program. I was so happy when Radhika started talking about EMT because I have some EMT stuff in my own research and I don't have to give that much of a background. I was actually worried last night about that. So, you know, I think that what I want to try to hopefully leave you with is kind of a little bit of an epigenetics journey. A lot of you probably don't hear about histone post-translational modifications, chromatin remodeling, other things like that. So hopefully everybody will learn something from the talk. Let me get my slides to change here. Okay, so the first thing I want to do, there's a lot of people that contribute to the research in my lab, but I have to point out one individual, Mike Wilson, who was a postdoc in my lab, who recently took a job here at Wayne State. He's now an assistant professor in the Department of Oncology. You'll notice that a lot of the actual papers that are my own papers that we published have Wilson et al. Listed. That's because he's the first author. He spearheaded a lot of the work. So I can't come to a talk and talk about chromatin regulation without talking about chromatin remodeling, because it's really an important process in the cell. The first evidence of chromatin remodeling came from the publication of these two papers, which actually occurred in the late 1970s by Harold Weintraub and Carl Wu. And what they were doing was they were studying the DNA accessibility using different nucleases, MNase or DNase-1, on chromatin for heat shock proteins after heat shock stimulus. What they found was that generally, when you have an inactive state, so in the absence of stimulus, this chromatin configuration was closed, and that's because these nucleases were unable to attack their DNA templates. However, when they added stimulus, the nucleases could attack the DNA. You would get a certain type of digestion pattern indicative of this open chromatin state. And so again, this was in the late 1970s. So it wasn't until really the 1980s that scientists started cloning a bunch of different yeast enzymes, looking for different phenotypes. One of the phenotypes in particular that was interested at the time was this ability to either switch mating types or utilize sucrose as a carbon source, or the switch SNP mutants. And it was later found through successive cloning that switch SNP was actually components of a large multi-protein complex that can remodel or reconfigure nucleosomes in response to ATP or through ATP hydrolysis, thus allowing co-activators, transcription factors, or co-repressors to actually access their DNA templates. So the largest subunit in the complex is ARID1A, which is the protein I'm going to be talking about today. This particular complex consists of between 9 and 11 subunits in mammals. So it's important to note that the process of chromatin remodeling doesn't involve just allowing transcription factors to bind their DNA templates. There are a lot of other processes in the nucleus that require chromatin remodeling. I'm showing two here. The process of histone exchange, or the exchanging of these nucleosomes on DNA, is very much coupled to chromatin remodeling, as well as the process of histone modifications. So a lot of these lysines and arginines and other types of side chain residues that are present on histones, they're buried and they have to become accessible. And so chromatin remodeling is really important for increasing the accessibility of these side chains to some of these histone modifying enzymes. So as many of you know, there's a couple different subtypes, really 4 major subtypes of ovarian cancer. A lot of people study high-grade serous because it's the most predominant. There are other subtypes that are strongly associated with endometriosis. So at least in a subset of cases of clear cell and endometrioid, one of the sources of these tumors is thought to be endometriosis. It's long been known to be a major risk factor for these cancers. It's considered a precursor lesion. And the idea here is that if you have menstrual fragments that would normally flow outside the body, if they would move in a retrograde fashion through the fallopian tubes, deposit themselves on the ovary, that there's some change or transformative process that occurs in that microenvironment, either in a cell-autonomous way or non-cell-autonomous way, that causes a subset of these ovarian endometriosis lesions to become ovarian cancer. It's true that when in cases of clear cell carcinoma as well as endometrioid, a lot of these cases have contiguous endometriosis. So you'll see clear cell carcinoma of the ovary coincident with atypical endometriosis. So one of the best kind of molecular evidence that we have that a subset of ovarian cancer can come from endometriosis comes from ARID1A loss. So in 2010, ARID1A mutations were first identified in endometriosis-associated ovarian cancers. ARID1A is also mutated in endometriosis. A lot of deeply invasive forms of endometriosis, such as ovarian, like endometriomas, but also deep infiltrating carry ARID1A mutations. Partial loss of expression is observed, but we also observe complete loss of ARID1A expression. It's important to note that ARID1A loss is often observed in this epithelial compartment of the lesion. And just shown here is a ARID1A wild type patient versus a patient that has a frame shift and activating mutation specifically in this epithelial compartment. And you'll notice loss of ARID1A immunoreactivity specifically in the nuclear domains of these cells. So the endometrium, as many of you already may know, is composed of both epithelium and stroma that can undergo cyclical changes in response to steroid hormones such as estrogen and progesterone, really in preparation for embryo implantation. My lab has been very interested in the epithelial compartment of the endometrium because this is where we see a lot of the tumor mutations. And specifically, we've been interested in both luminal and glandular components of the endometrium. Just shown here is a cross-section through a human endometrium, and you can see this glandular epithelium, and then you can see the luminal epithelium up here based on the actual lumen of the uterus. So just to provide a little bit of a summary and bullet points before I start getting into some of the data, it's known that these inactivating ARID1A mutations result in loss of protein expression. In both benign and malignant disease, ARID1A loss alone is often not sufficient to cause disease. We know based on animal models and also just the co-occurring mutation patterns that occur in a lot of these human tumors that ARID1A mutations are often coexist with mutations that cause activation of the PI3 kinase pathway. So we often will see ARID1A loss and PI3 kinase pathway activation in these tumor cells. It's also important to note again that this epithelial loss of ARID1A is really important. A lot of our models are based on epithelial loss of ARID1A. This includes both mouse models as well as cell-based models. So when I started my lab, we were really interested in trying to understand the role of ARID1A specifically in the endometrial epithelium. We turned to our mouse models. So a mouse model that we've used quite a bit of in the lab is shown here. This particular model relies on inactivation of ARID1A or activation of this ROSA26 PIK3CA transgene. So this particular transgene contains the human cDNA for PIK3CA knocked into the ROSA26 locus. This particular cDNA contains this H1047R hotspot mutation. And so in the presence of Cre recombinase, you're going to get loss of ARID1A or activation of this PIK3CA transgene. So the Cre allele that we use a lot of in the lab is called lactoferrin Cre. This particular Cre is very specific to the endometrial epithelium. It turns on as the animals first initiate estrus. So it comes on naturally in adult mice. And just shown here is LAC-Z expression showing this domain of Cre activity in the epithelial compartment. So one of the first things that we noticed in the lab when we started doing these mouse studies was that ARID1A loss was really associated with epithelial mesenchymal transition, specifically in this epithelial compartment. And so we did a lot of RNA-seq studies. Just shown here is a heat map on the left, showing a group of EMT genes that were identified through the Mack and Tong pan-cancer EMT gene signature paper. And you'll notice that in these ARID1A, PIK3CA, lactoferrin mutants, you can see loss of these epithelial markers and a gain of mesenchymal markers. That's more clearly shown here in the immunostaining to the right. So if you'll notice, on the top panels is E-cadherin staining, or CDH1. And the bottom panels is vimentin, which is our mesenchymal marker. In control samples, E-cadherin staining very clearly marks those adherens junctions that are staggered throughout the entire epithelial layer. With ARID1A loss, you can see loss of E-cadherin expression in these cells. When we add in this PIK3CA mutation, we see endometrial hyperplasia in these mice, but that's also coincident with this disorganization of this epithelial layer and loss of E-cadherin. You'll notice if you look at this mesenchymal marker vimentin, in normal control samples, vimentin is very strongly associated with that endometrial stroma and doesn't really bleed into that epithelial layer. But in ARID1A mutants, as well as our double mutants containing the PIK3CA transgene, you'll notice that you can see cells start to express vimentin in the cell layer. Hopefully everybody can see that. So the other really important piece of evidence that really connected a lot of our work back to endometriosis was what I thought was a very elegant study that we didn't think was going to work at all. But we did it anyways, because we knew that these abnormal endometrial epithelial cells that were present within the eutopic endometrium were undergoing EMT. They were becoming invasive. They were gaining all these properties that Radhika so elegantly talked about. And we reasoned that if we created an incision right at the uterotubal junction of the mice, that we would expose the lumen of the uterus to the peritoneal space. Now, the reason to do this is because rodents have a very long and convoluted oviductal network that's surrounded by a bursal membrane. So there's no natural way for these luminal cells to access the peritoneum. So essentially what we were doing was we were removing this anatomical barrier of the oviducts and the bursa. And I've just shown a few whole mount images here of some of the phenotypes we observed. About 40% of the mice developed ovarian lesions. This included cystic ovaries. Importantly, the actual cystic features were found on the ovary where the ovarian or where the uterotubal incision occurred. So the contralateral control did not show these phenotypes. These mice also developed endometriotic-like lesions throughout the peritoneal space, both in the omentum as well as on the intestines shown here in these arrowheads. For these studies, we incorporated a GFP tracer, which is Cre-activatable as well. And so we can trace these mutant cells as they migrate throughout the peritoneal tract, peritoneal region. And you'll notice at the bottom here in these, the uterotubal incision ovary or side of the uterus, these GFP positive cells kind of coalesce around the ovaries themselves. And we don't see that in the contralateral controls. So this was kind of like in vivo evidence that the cell autonomous feature of invasiveness was really allowing these cells to migrate into the peritoneal space and survive and attach themselves and grow in this area. And so, through a lot of work from Mike and others in the lab, we settled on this idea that ARID1A loss was causing these cells to become invasive. As Radhika noted, invasiveness means a lot of things. It's not just becoming more mesenchymal-like, it's degrading basement membranes, being able to migrate, undergo anchorage-independent survival, as well as altered cell adhesion. So what this suggests is that normally when ARID1A is present, it's maintaining proper endometrial epithelial cell identity. It's allowing those cells to stay epithelial-like. And so what we wanted to really know was what were some of the epigenetic mechanisms that were driving this change between an endometrial epithelium to this more invasive endometrium. And so our central hypothesis all along has been that ARID1A loss in this tissue leads to epigenetic regulation that would be associated with acquisition of these invasive cell properties. So I just want to switch gears a little bit and start talking a little bit about some of the approaches we use in the lab. We do a lot of genomics, a lot of RNA-seq. Every study that we do probably has some RNA-seq data set associated with it. We also use ATAC-seq, which allows us to measure chromatin accessibility. And we can do ChIP-seq as well as cut-and-run-seq for various histone post-translational modifications that allow us to assign function to the genome through this epigenetic correlations with function. And so to kind of give you some, just a little bit of a epigenetics 101, so H3K27 acetylation is really an acetyl group on lysine 27 of the tail of histone H3. This particular mark is associated with active regulatory regions. If you have a methyl mark on that same lysine 27 residue, you see associations with repression. And so we can use these patterns of these marks across the genome to assign function to the genome, to the genome. Just shown here is a UCSC genome browser plot showing some of that data in raw form. There's two genes here that are expressed. You can see RNA signal coming from these loci. You can see these two genes are coded with active regulatory elements. However, this gene on the far right, which is not expressed, is coded by this H3K27 trimethyl mark. So a lot of our studies, we've used a endometriosis cell line called 12Z. And this is a T antigen transformed endometriosis cell line. We have validated a lot of our studies and have done a lot of studies in vivo as well. So we don't always rely on 12Z, but oftentimes these cell lines are our initial kind of entry into a particular question that we're trying to ask in the lab. So one of the things that you can do with a lot of these different features of the epigenome is build chromatin state models, which are statistical models that allow you to segment the genome into or compartmentalize it and then assign function to it. In this case, I'm showing you an 18-state chromatin state model showing all these different features. So we've assigned these through empirical data, but this is our own assignment. We're saying this is polycomb repressed because it contains H3K27 trimethylation, but doesn't contain any of these other active marks. But these states are derived through empirical data in cells. And the important thing for a lot of our studies is we often have looked both before and after ARID1A loss. So we're looking at acute loss of ARID1A and asking what's there before and then what happens after it's lost. So we can use these chromatin state maps, if you will, to look to see how these changes are occurring when ARID1A is there or when it's lost. So one of the first things we wanted to ask is, based on our 18-state chromatin map, where is ARID1A actually bound? And so we had ARID1A ChIP-seq data. We can compare that ChIP-seq data to these various chromatin states, and you'll notice that ARID1A is specifically enriched at this class of enhancers called super enhancers, which is shown here, as well as highly active enhancers. So it's associated with highly active regions of the genome. So I just want to introduce the concept of super enhancers to this audience. These are basically clusters of enhancers. They often are very large. They can be anywhere from a kb to about 100 kb in size. They bind multiple different transcription factors and really integrate signals, multiple signals in the cell. We know that based on data from development and developing tissues, that genes that have super enhancers are often your cell identity genes. They're the key transcription factors or signaling molecules that are really important for shaping tissue identity. And so it made sense to us if ARID1A is regulating cell identity, that it might be found at super enhancers. So again, a lot of our profiling is done before and after ARID1A loss. So one of the first things that we observed with loss of ARID1A in these 12Z cells was that we see a lot of change across the epigenome, but one of the major features that we saw was changes in H3K27 acetylation. Shown here are MA plots. So what you're seeing is ChIP-seq signal either going up or down with ARID1A loss. This is kind of like what you would see with like a microarray or something like that. And you'll notice for comparing ARID1A SH treated cells to control cells, you see a lot of change in H3K27 acetylation. About 21% of sites are changing with ARID1A loss. This is a global change. But you'll notice this other active marker, there's really no change. So it's very specific to H3K27. It's a minor fraction of sites are changing for H3K18 acetylation. So this really got our attention to focus more on H3K27 acetylation. Importantly, when we started to look more closely at some of these highly active regions, either promoters, enhancers, or super enhancers, it was at these super enhancers where we were seeing an increase in H3K27 acetylation with ARID1A loss. So normally when ARID1A is there will be low levels of H3K27. When it's lost, H3K27 acetylation goes up. So based on these findings, we knew from the literature that the histone acetyltransferase that actually deposited H3K27 acetylation was called P300. This particular enzyme is really important for adding, it's not the only enzyme that adds this mark, but it's really important for adding H3K27 acetylation to these nucleosomes. And we reason that if we're seeing this upregulation or this increase in H3K27 acetylation super enhancers in the absence of ARID1A, that P300 may be actually adding that mark, and it may create this dependency. So we wanted to ask, is ARID1A deficient cells dependent on P300? And so we turned back to our cell-based model, which allows us to measure invasion. This is a really simple transwell invasion assay where we plate 12 Z cells that have been transfected with siRNAs or plasmids onto matrigel-coated transwells. We let the cells invade the matrigel, go through the transwell membrane, we scrape off the inner cells, and then we stain those outer cells with calcine AM. And so any cells that have gone through the membrane and through the matrigel are going to be stained green for calcine AM. And you'll notice here you see increased staining with ARID1A loss. So to tease apart this dependency on the actions of P300, we co-transfected 12 Z cells with their siRNAs targeting ARID1A or P300. And you'll notice that we see this really nice response where we see increased invasion with ARID1A loss. But when we inactivate P300 in this context, we can see reduced invasion, suggesting that P300 is driving invasion in these ARID1A deficient cells. So we next turned to a more pharmacological approach to inhibit P300, focusing on a drug called A485, which is a drug that specifically targets P300 HAT activity. So this is going to block enzymatic activity of P300. Like the P300 siRNA experiment, we went back into our 12Z model, only now we're treating with increasing concentrations of A485. And you'll notice that even small amounts of A485, 10 nanomolar, are capable of significantly inhibiting ARID1A mutant cell invasion in the cell model. We know that a lot of these concentrations are sublethal, so we didn't see any effects on cell health. So this kind of brings us to this point where our model was that in the normal endometrial epithelium, switch SNF was bound to super enhancers, and it was antagonizing P300 HAT activity. In the absence of ARID1A, this repression will be relieved, allowing P300 to come in here and hyperactivate these super enhancers by adding these acetyl groups. And of course, this could be inhibited by A485. But we had this great phenotypic model. We knew that all of this was associated with invasion. And we really wanted to understand what were the invasiveness genes that were driving this change. And so we turned back to genomics. This is a very complicated experiment, and it worked really well, so I can talk about it, but often these don't work that well. This is actually our first foray into cut and run, which is basically a technique which is similar to ChIP-seq, only the antibodies are conjugated or tethered to MNAs. And so the idea here is that these antibodies are going to localize to these K27 acyl residues throughout the genome. The proximity of MNAs to these sites is going to cut these regions, and of course, cut DNA can be tagged very easily by Illumina-based sequencing, and we can map those sites back to the genome for abundance. We tested all the different combinations, both ARID1A loss, P300 loss, drug only, or ARID1A plus P300 and ARID1A plus drug treatment. So naturally, you're going to see a lot of different changes happening. Just shown here is comparisons between ARID1A versus control. ARID1A loss versus cells that are also depleted for both ARID1A and P300 or treated with drug. Naturally, again, you're going to see a lot of different changes. There's this group of genes where acetylation was basically going down globally in all conditions. You had these regions where you saw it going up, but then what we were interested in was in these gain reversal genes. So these were going to be factors where we're seeing an increase in K27 acetylation with ARID1A loss. But if you removed P300 or inhibited its HAT activity in that context, you would see loss of K27. So we refer to these as gain reversal for just that reason, and we thought that these gain reversal genes would allow us to more finely map where those invasiveness genes were in the genome, or what regulatory elements were driving this change. So these would be our genes that would be implicated in ARID1A mutant cell invasiveness. So we needed transcription data. So we went back in, we did the same treatments, same drug combination, same siRNA plus P300 plus drug, but this time we did RNA-seq. And so the idea here is that we were looking for genes where we would see them go up with ARID1A loss, and that upregulation would be suppressed by P300 inhibition. And so naturally, you're going to see a lot of change. This is a very busy slide. Hopefully everybody can see it. But shown in this Venn diagram on the left is those same comparisons that were made in the heat map on the previous slides. Only we were most interested in these genes, this 138 genes in this triple intercept class. These are going to be genes that were affected in these conditions where we thought they would be driving invasiveness based on these comparisons. So shown here is a heat map of all those 138 genes. We're looking for genes that are going up with ARID1A loss and down with either P300 loss or HAT inhibition. And if we now further break that down, now we have 50 genes in that grouping. And we start to ask questions like, did they have a super enhancer that was in this gain reversal category? And were they regulated by a super enhancer in normal conditions? We basically narrowed it down to one gene, and that gene was called SERPINE1. And so what is SERPINE1? This is also called plasminogen activator inhibitor 1 or PAI1. Normally, SERPINE1's function is to, or the canonical role of it is to inhibit the plasminogen activator pathway. So you would expect in a situation like an ARID1A deficient cell where you see increased SERPINE1 that you would see increased fibrin accumulation in the tissue itself, because this is going to block this fibrinolysis of this fibrin. However, it's known that SERPINE1 also plays roles in integrin signaling through its roles at the membrane. These non-canonical roles are thought to be really important drivers of both cell migration as well as invasion. We wanted to ask whether or not SERPINE1 was actually required for ARID1A mutant cell invasion. So we went back to our cell model, depleted the cells with ARID1A as well as SERPINE1, and found that in fact SERPINE1 loss abrogated this ARID1A deficient cell invasion as expected. So we also, throughout all these studies, we were very cognizant of our in vivo physiological relevance, turning back to our mouse models. Just shown here is a series of sections to a mouse endometrium and control. Animals that have P300 loss in this epithelial compartment have our double mutation in either ARID1A or PIK3CA, or now a triple mutation where we're inactivating P300 in this context. And you'll notice SERPINE1 is being upregulated in this epithelial compartment. And that's clearly dependent on P300 because you don't see that upregulation here. We also know, based on our endometriosis model, that SERPINE1 is upregulated in those ectopic endometriotic lesions as well. So we were excited to see SERPINE1 because it's known for a while that SERPINE1 was implicated in endometriosis. It's known to have higher expressions in ovarian endometriomas, as well as deep infiltrating endometriosis. There's some recent work from Ronnie Drapkin's lab showing SERPINE1 playing a role in high-grade serous, in particular angiogenic processes there. We haven't explored that in this context, but I think there's definitely a precedence for studying SERPINE1 in these contexts. So with that, I'll go ahead and take any questions. Again, I really appreciate the invitation to present my work today. Thank you.
But when you measured certain one over super cancer, was it all through the super cancer or localized to spontaneous?
Was it all through the SERPINE1 enhancer? Yeah, so the interesting thing about, I wish I had, I typically show a locus shot of the SERPINE1 super enhancer, but there's definitely domains of K27 within that super enhancer. And you can break down, the SERPINE1 super enhancer is literally 50 kb in size. And within that, there is micro domains in there. Those could be, regulatory elements that have transcription factor binding sites or other things like that. We don't really know. But one of the things that we have observed with a lot of our work is a lot of the super enhancers that ARO1A regulates, they're actually super enhancers that contain promoters in them. So they typically are very large and they're considered super enhancers, but they actually, those super enhancers bleed into genic regions. And in the case of SERPINE1, that super enhancer does bleed into the SERPINE1 gene itself.
One other thing, have you ever extended your study to start looking at three-dimensional structure? Because right now you're looking at it as a plan. And obviously there's other contacts that are going to miss something.
Yeah, I've wanted to get into the 3D kind of space, looking at that. I think those are very relevant, especially in the context of some of the things we're seeing. I think one of the challenges we have is just finding the appropriate models. We're really trying to leverage more in vivo models in our approach and stuff, and we're just not at the economy of scale where those types of methods can be easily utilized in an in vivo context. I'm hoping maybe with technology we can start doing those, but it's just more cell line work, and it's just something we haven't really approached. There's also cost and just there's some technical aspects as well. That's the obvious next direction where we would need to take this for sure. So
if you noticed any relationship between your observations or between the patterns of methylation or acetylation and phase of the estrous cycle?
That's a great question, actually. We have some unpublished data suggesting that we do see changes over the estrous cycle with some of these marks. We've been very interested in variant histones in that regard, not necessarily methylation or acetylation, but this idea of these variant histones being swapped in very dynamically throughout the menstrual cycle. And we've gotten some newer data on estrogen target genes being bound by a particular variant histone called H2AZ. We have data now to suggest that the histone exchanger that deposits that actually plays a role in estrogen gene regulation as well. So
because some of these observations may be relevant to physiological status of how endometriosis occurs either in the absence of an ARO1 mutation, but also
absolutely
steps in endometrial cancer, right?
Yeah,
because driven by other factors, right?
Absolutely.
And then relevant to that, if you notice in your work that, do you find that the endometrium is characterized by a purely epithelial phenotype, or do you see markers of, like, say, that might be characteristic of stroma in the endometrial epithelium that really shouldn't sort of belong there?
Yeah, we could probably have a serious beer conversation about this, but I think that, the endometrium is very dynamic and it changes a lot. And I would say that, even in it's different between species. We can't always rely on a mouse because mice don't menstruate, and they're very different than humans. So I think there's a big black hole as to what's going on across the menstrual cycle in that regard. From a cellular level, we know that there's definitely changes. You go from a secretory epithelium that's a protein pump making mucins and stuff that are allowing those embryos to implant to a more proliferative epithelium, and there's so many different changes that are happening there. I think in the context of a lot of these diseases we study, you have to think about the window of exposure and the window of opportunity for these cells to become malignant or maybe become disease. And in our case, endometriosis is very much a disease. It's a disease of menstruation, right? That's the idea.
Well,
if you're a believer in that,
yeah.
But so maybe we need to be looking in that window of time more closely at some of these molecular changes that are happening. But I will say, from an epigenetic perspective, it's very dynamic. Absolutely. So
I just want to add one thing. I know there is a question and there's a quick comment about your model of the migration and so on, the fallopian tube. So we worked for many years trying to understand what is going on. We can put cells. If you super ovulate those animals,
you will have penetration going inside of your implants. And it doesn't matter, you don't need even to cut the fallopian tube. It will go through the fallopian tube and the ovaries. We can talk more, I'll show you that.
Yeah, sure. We've stayed away from hormone supplementation because
I know.
a lot of the pre-alleles that are really useful for studying endometrium, they're all hormone responsive genes. So as soon as you start adding hormones into the mix,
You
or
change-
cutting out over, you change your genetic model changes.
But at least for the migration and the semen
Yes,
that's going
yeah.
to the ovaries, that's it, so.
Yeah.
You have a question over Zoom.
How do we?
If Phil.
Can you hear me?
Yes, we
Yeah.
can.
Just apology that I am a neophyte in this area, but what your discussion was very exciting, just in terms of this invasiveness model. And I'm just wondering if for any of the other people in the room who are more studied the demographics of women getting ovarian cancer or responses to treatment, if this has lit up anything for them in terms of what they've seen or wondered about, or as a possible pathway. I don't know if that makes sense, the question even.
I understand what you're asking, and I don't know if I'm the best person to answer this, but I don't know if what I'm showing is highly correlative of invasive metastatic ovarian cancer. And I'm more thinking of the precursor lesions that are happening before ovarian cancer could occur. So this idea that these endometriotic cells have invasive properties that are cell autonomous, that are cell intrinsic, that's allowing them to survive if there is a retrograde menstruation event or something like that, if they acquire an ARID1A mutation, for example. But I'm not sure that answers your question. Go ahead, Annalise.
I just, have they done enhancer profiling of stage 4 endometriosis tissue that have progressed to, let's say, cancer, and those that have not? And if so, have they found similar super enhancers that you found? Or have they done gene expression and has Serpine or PA1 been one of the hits?
That's a great question. I would say those studies have not been done, or at least not published. I think it would be really challenging to do those studies just with the technology and access to tissues and other things like that. But I will say that in our limited deep dive into super enhancer function in the mouse endometrium using cells that we've isolated and stuff, a lot of super enhancer containing genes are actually estrogen targets. So a lot of these hormonal targets that are normally, we think of estrogen as being the identity factor for the endometrial epithelium in many ways. It's driving that identity. And a lot of the estrogen targets have these super enhancers. So from that kind of idea, you think about endometriosis being an estrogen dependent disorder in many ways, then that kind of all jives. We don't have direct evidence in the lesion.
But you could go back and look at, I don't think it would be too difficult to actually collect a series of endometriosis-associated ovarian cancers and look to see if you're seeing some of the same methylation marks in those carcinomas.
Yeah.
Or even in specifically deep endometriosis.
Yeah.
There's not a lot of similarities also with the clear carcinomas.
Yeah, well, I mean, yeah,
Clear cell carcinomas.
those also arise in the same region.
Exactly.
Yeah, I think
I mean,
we would probably have to make sure we went back, and I know, like in West Michigan, I know how many clear cell patients we've had.
Yeah,
in West
that's true.
Michigan.
It's more difficult.
It's so, I mean, you could never design a prospective study for that. It'd have to be something that's very collaborative and probably even archive tissues.
As Dr. Morris, he's here.
Yeah, but being able to do these types of chromatin-based studies on fixed tissues and stuff, I don't think the technology is we're not there quite yet, where it's easily accessible by everyone.
or flash frozen.
That might work, yeah. So,
How many of the ARID1A mutated endometriosis, what's like, actually, result in cancer? Like a small percentage or a large?
so a lot of the studies where they've seen ARID1A loss in these endometriomas is in contiguous type situations where the patient has clear cell, there's loss of ARID1A and the adjacent.
50% of
About
the endometriosis-associated
yeah,
cancers have ARID1A loss. but
yeah. So about
what is-
50% of those have ARID1A mutations, basically.
No, but
that's-
of the endometriosis that has the majority with transformed to cancer or?
I don't think so. Because I just don't think so. I think that there's enough data, even, you know, AR1A is mutated in like 10% of deep infiltrating. We've seen through unpublished studies that it's downregulated in peritoneal endometriosis.
But
Those don't become
most
cancer.
of them
Yes.
don't become cancer.
That's right. Yeah. So I think that's a really good point because a lot of the things that we're seeing is probably necessary, but not sufficient for malignancy.
But,
Yeah.
but your model system is driving the overexpression of PI3K, right?
Yes.
So that's another major hallmark of endometrial or carcinomas arising with an endometrioid phenotype.
Yes.
right?
Yeah, I think the thing to keep in mind is those models also develop endometrial hyperplasia and vaginal bleeding, which is not seen in endometriosis ovarian cancer. So the mice are kind of like mutts. They have like a little bit of everything in terms of the phenotypic spectrum. And I think if anything, our mouse model and all the data we've shown targeting endometrium is that the ovarian microenvironment is probably one of the most important features here because we're putting those mutations that we see in the tumors into the mice, and we're getting this kind of spectrum. We're not getting, you know, all the mice develop, you know, ovarian cancer, and that's it. There's other things going on. We know from the clinic that endometrial cancer is very different than ovarian cancer. And
But
so many-
not in terms of, not necessarily in terms of the types of drivers that you see in endometrioid adenocarcinomas arising in endometriosis. Like those look more like endometrial cancer.
That's true. They do.
Right?
Yeah.
Okay, and it seems to me like you have a model that generates something of an intermediate phenotype. Yep, it's an
Yes.
There's all that biologic difference. But it seems like it would be very helpful for taking what you're doing and then using, kind of moving that forward to understand these cancers to try to see if you can bookend this, if you're where it relates to that spectrum, even if it's some relatively something simpler,
Yeah.
like comparing these patterns to, say, endometrium, which has a lot of mutations at baseline, by the way. Okay, you can see AR1 mutations in healthy endometrium. Okay, and then endometrial
Right.
hyperplasia, endometrioid adenocarcinoma. Right, and see where does your model slot into this, right?
Yeah.
To me, it just seems like it would be helpful to understand what you're sort of you're studying, how they're relevant to the process.
Yeah, you're the kind reviewer.
I think it's interesting.
Okay. Thank you. Oh, what we got another?
So, Matt, I like where you're going with this, that and that is going towards, and clear cell and endometrioid ovarian cancer are relatively uncommon.
Right.
And with those, I think it's a nice mechanistic model, but I think what we do see fairly commonly are those endometrioid cancers that are PI3K, ERG1A mutated, that are deeply invasive in the endometrium that we really don't have good explanation
Right.
for to begin with. And we've just restaged, we revised the staging for endometrial cancer, and it's much more molecularly classified, if anything else. And I think when we look at this, this is really interesting to see if it really does correlate to those deeply invasive grade 1 endometrioid cancers we see in the endometrium that we don't really have a good explanation for otherwise. They're not supposed to behave that way.
Yeah, we'll say from a paper that we have published, we have shown that the AR1A PIK3CA mutant endometrium has gene expression features that look like invasive endometrial cancer based on TCGA comparisons, looking at just RNA expression. So that is there. I think maybe we should go on break. But I think that the different, it's really important to think about the difference in the diseases though, because I do think there's risk factors that are found in endometrial cancer and not ovarian cancer and all these other things. And once you start to bring in all of those different risk factors and stuff, they really are kind of distinct diseases, even though they have the same kind of molecular fingerprints, if you will.
Maybe we'll talk right away.
Yeah, let's talk about it.
And you know, there is a panel discussion also for the public at the end of this. So you're more than welcome to stay and have that conversation.
Sure.
And there is a break after one more lecture.
Thank you very much, Ronald.
Thanks.
Thank you for that fantastic talk and very productive discussion. So I'm going to introduce our next speaker, who really do not need any introduction.
Up to
Dr. Saed
5 minutes.
is an associate professor in the Department of Studies in Oncology here at Wayne State University. He is also the president of the National Arab American Medical Association in Michigan. His primary research is focused on pathogenesis of tissue fibrosis and the role of oxidative stress in the pathogenesis of epithelial ovarian cancer. His research has identified and pulled several patents on novel biomarkers for early detection and novel therapies for ovarian cancer.
Thank
Dr.
you,
Said.
Aisha. Thank you. 5 minutes, and we'll go to break. So good afternoon, everybody. Thank you for coming. I would like to thank our sponsors, all of them, especially Mass Mutual, my friend Adam. Thank you. Okay, so we have many projects going on in our lab right now. I like to share with you one project that actually made it from the bench all the way to patent, to license the patent from Wayne State to right now clinical trial. So might be very short. Okay, don't ask me too many questions. Okay, so I'm going to skip this. All of you know ovarian cancer. So the story of this patent started when we discovered that epithelial ovarian cancer cells express a myeloid protein known as myeloperoxidase. So myeloperoxidase, this guy, is from the name, you can tell, it's only found in myeloid cells, blood cells. No one had ever reported this in a non-myeloid cell. So this paper, I think we were the first to report it in 2010. Okay, long time, 23 years ago. After that, we also published with our group that if this could be, this myeloperoxidase could be a potential biomarker for early detection of ovarian cancer. At that time, we didn't know what we were doing, but we were asking one question: why epithelial ovarian cancer cell need to make its own myeloperoxidase? It is abundant in the blood, right? So why making it inside? We didn't have the answer to the question. Years later, after us, 4 years, 2014, 2016, other people confirmed our finding, and that it made us more encouraged to go on with this line of research and find out what is the meaning of this expression. What does it mean? Why the cells need this? Okay, so this is where we started. Myeloperoxidase is a heme protein. It's made from 2 polypeptides. They are from the same size, 75 kilodalton. So this is one single, 75, 75. And they're linked together by a disulfide. So 2 monomers, peptides, 75 kilodalton each. That's very important because this depends what I'm going to talk about in a minute. And they're linked together by a disulfide. Okay? We found that it is expressed in ovarian cancer cells, little expression in other cells, other cancers, other than ovarian. So, the interesting part, recently we found something really puzzling, that in healthy serum, you will find 75 plus 75 is 150. You will find the mature dipeptide myeloperoxidase in the healthy serum. In ovarian cancer serum patients, you see the healthy, the dipeptide, the 150, but also you see the monomer, the 75 kilodalton monomer peptide. And further testing with these ovarian cancer cells, we found that they only express, these are cancer cell lines, COV, 8.780, NDH, those are ovarian cancer cells, cell lines, and they only express the monomer. That was really surprising to us. So we didn't know what it means. But we collaborated with the Center of Molecular Medicine and Genetics. They gave us some samples from Dr. Tanski's lab. And they gave us some plasma samples where we looked at and tested whether this monomer can actually discriminate between early stage versus the rest of the stages. So we want to know, not the rest of the stages, sorry, that's the second question. The first question is whether it will discriminate between healthy and early stage. We don't really care about after that. So it looks like, if you look at the monomer, this is the control, healthy individual. These are non-cancer inflammatory gynecologic conditions such as endometriosis, fibroids, all of that stuff. This is early stage, which is stage 1 in this case. This is the rest of the stages, all pulled together. And you can see how monomer discriminated easily between them. And also the dimer failed to, when we compare late stage with early stage, the dimer couldn't do it, but the monomer did. So now if we patent this, and now this is being tested on many other samples that we identified. And the results so far is really encouraging to find, as you all know, we lack a very specific early detection marker for ovarian cancer. So this could be the one. So now, why I'm talking about this? Because this is what brought us to the discovery. So now we wanted to know, what is the significance of this? Again, why ovarian cancer cells, epithelial cells, express myeloperoxidase? So the answer to this answer led us to search in the literature. We found a paper that is published in PNAS, I think, or Journal of Science. They say that there is a ligand to myeloperoxidase. So myeloperoxidase is expressed in macrophages. And if we get wound or infection, macrophages are called in to the site of injury, and they wear protective clothing so they can go on to extinguish the fire. The protective clothing is myeloperoxidase binding to its own receptor, which is called CD11B, CD18, with another name for it, beta-2 integrin. Okay, so this binding actually made the lymphocytes, the T cells, are more resistant to apoptosis, so they don't die, they can go do their job and leave. So we said, okay, well, maybe this is the situation here in ovarian cancer. Let's check it out. Easy. Let's determine CD11. Is it there or not? So we looked at CD11 and we found CD11 staining in normal ovaries, more in the cancerous ovary. It is also, that's the positive part, on other cancers. Is expressed in lung, colon, endometrial, liver, so it's everywhere. When we use the antibody against CD11, we find that it has a cytotoxic effect. So CD11B, 5 micrograms, 10 micrograms, dose response, increase the killing. And this killing is very specific. So if you use an isotype of the CD11B antibody, you don't kill the cells. It doesn't touch or kill non-cancers. Too good. Too good. Too good to be true, right? So we said, okay, what kind of killing is this? Is it apoptosis? So we did cascade 3 with the tunnel assay. It is apoptosis. Many reviewers say, no, it could be endometriosis, where this binding of the cells, attachment of the cells to the cell culture plate actually killed them. So we did endometriosis assay and we found no difference between coated plate versus uncoated plate. So it is true killing through a mechanism of some kind of apoptosis. So now we said, okay, what we did, we immunoprecipitated total proteins from ovarian cancer cells with CD11 antibody. Then we hybridized with myeloperoxidase to see if there is a binding between the two in these cells. And you can see there is binding. We did the opposite, where we did immunoprecipitation with myeloperoxidase and probe with CD11B antibodies. And we found there is a relationship, there is a binding between the two. So what we did, we sent this So we have the immunoprecipitation by CD11 antibody. We sent it to mass spec here, Wayne State Core Facility. They actually, I thought, when you precipitate, immunoprecipitate with a CD11B monoclonal antibody, you should get from the mass spec project, you should get one band, right? Maybe two, maybe three. We got 300. So it's many, many interactions. What is going on? So when we did the CD, the mass spec, we found that
Okay.
Yeah, the problem is.
there's no,
Somebody has to put it there.
20 minutes ago, but I don't always look at it.
priority is given to people here.
We're asking the Zoom audience to please mute themselves. Okay, I think that. Thank you.
Okay, so what we did the mass spec, as I told you, and found no CD11. B or the partner, the AD. So what is going on? We detected this guy, LMTK3. When we did we took a sequence from LMTK3, 100 amino acids. We tagged them with GST. We run them on a gel. We actually found that the antibody to CD11B cross-reacted with this guy, LMTK3. And this is our target. And I'll show you. We also have other targets. As I told you from the list, when we start rerunning the list, coming down, bringing the list down to 1, which is us, 60 kilodalton each shot protein, heteronurin, HRNR, and titin. Also, B, alpha B, and beta 1. Now remember those four. Those four are really linked together with a mechanism with LMTK3. They're all linked together. And I'll explain it to you. The other 3-4 projects that's going on in the lab is identifying the role of these and how they interact with LMTK3. Now, LMTK3, I never heard of it in my life before, but then I started to work on it, study it, see what's going on. It looks like it's a tyrosine kinase with many signaling functions. It's signaling, influence proliferation, apoptosis. In the literature, any function I looked for, it is there. It is done by LMTK 1, 2, and 3. They are a family of 3 isoenzymes. They are localized in the nucleus, in the cytoplasm, in the intracellular membrane, and outside. They're released too. So they're everywhere. But the interesting part, that's what we found, that makes this really interesting, and people and this company got interested in this, is that in normal situation, LMTK3, the 3, not the 2, not the 1, is only expressed in the nucleus. And when the cells start getting stress, I leave you to define stress, okay? Start releasing this from the nucleus.
Yeah. Okay, I'm coming. You want to come here or do you want me to come there?
Don't come here.
Oh, sorry.
Okay, so where were we? Yeah, so we were, so the interesting part of this thing is that LMTK3 is expressed in the nucleus, in the normal cell. And when the cells are stressed, diseased, for any reason, they start expressing it in the cytoplasm, in the membrane, and outside. So this is the interesting part of those. So this guy is the expression of the cytoplasmic is correlated with tumor grade and cancer patient survival. And this has been done before us, but with different cancers. Breast cancer, colorectal cancer. Remember I showed you the staining with different cancers? It is there, okay? So we don't know the function of this protein in ovarian cancer yet. We have some idea, but this is the interesting part. This is really interesting part. So you don't need LMTK3. if you knock out an LNTK3 expression in the cytoplasm, in the membrane outside, you kill the cancer cells. You kill the cell, whatever cell that is producing it outside the nucleus. But in the normal cells, because it's in the nucleus, you don't affect the normal cell. This is really wonderful news. So we started to continue with this, and I don't want to go through the chemistry of this. Chemistry is boring. Okay, let's talk about. So here is the story. When we started, so I thought we have something really interesting. Let's patent this. So I went to Wayne State Commercialization Office. I'm so excited. Look at the data. What do you mean? This is good. Look, it's really interesting. So it took us 7 years to patent this. So when we patent finally this, I thought that we can patent the antibody that target LNTK. So unfortunately, or I don't know if it's fortunately, we couldn't patent the antibody because the antibody is published. It is there. It is already sold by pharmaceutical companies. They sell it. It's there. So what do we patent? I want to patent the idea. What idea? There's no idea. There's a target that if you target it, you kill cancer cells. So anyways, to make a long story short, they told us to find a different way to target LNTK3. So we collaborated with a scientist, a bright guy who was here in Carmanos, and then he moved to, I think, Arizona University for Phoenix. This guy said, okay, I can make peptides for it that can target the LNTK3 and inhibit it, shut it down. Okay, let's do that. So he did something. I'm blocking the sequences of the peptide because they asked me to do that. Okay, so these are about 15 different peptides with different affinity to LNTK3. They were generated from, they have a peptide array of 18, 16 mer amino acids linked together, about 330,000 of them in a library. And they actually mapped the LNTK3, the protein itself, and found out where are the sequences, pick these sequences of peptides that actually bind LNTK3 and act like an antibiotic to block it. And this is a study. They asked us to find if there is affinity, what's the affinity or dissociation constant of these peptides to the target, the receptor, the LNTK3. And we did that. And to also make sure that in vivo, does it really bind LNTK3 or does it bind somewhere else? How do we know that? We did experiments where we just precipitated a long protein in a western, hybridized with the antibody, hybridized with the peptide, and you can see you get the same size band on a western. If you do the immunoprecipitation, this is with the antibody, this is with the antibody and the peptide. You can see when they're merging, you see the yellow, which means that the peptide, the antibody are binding to the same spot, whatever that spot is. Hopefully it's LNTK3, not where somewhere else. Okay, so we got those peptides. Those peptides are patent right now. And those peptides, we use them to see if they have a cytotoxic effect. And we compare to the cytotoxicity to the antibody. So this is the antibody of LNTK3, 5 and 10 micrograms. And this is the peptide. We picked the first two peptides with the highest affinity from the table that I just showed you. And also we use 5 and 10 micrograms. And we found that indeed there is a killing, and the killing is indiscriminate. It will kill resistant cells, sensitive cells. It will not kill macrophages, for example, which we expect. This killing is actually synergistic with chemo, so it will use a very low dose of chemo with peptide or with the antibody. If you knock down siRNA, use the siRNA to knock down LNTK3, you can see that the normal cells in yellow, see the yellow normal cell? Nothing happened because they don't really express it. It's in the nucleus, it doesn't go outside. But with the other ones, you can see with the other ovarian cancer cell lines, you use 10 nanomolar, 20 nanomolar, 40 nanomolar, you can see knocking down L1TK3 in a dose response manner. The interesting part, this is SPID mice, and they grow the tumor size shrink when you have the peptide. This is, in my opinion, the most important thing that we need to, and we are actually this, I will tell you in a minute that all this work, of course, the company before they licensed the work, you think they believe me? They did everything on their own. They have 3 different centers in Europe, one in Sweden, one in Spain, and one in Switzerland. And they actually run all this work. And they reproduce everything, and I'll share with you some of the data that we got. But this is Taxol, I think. What is it?
Is it
I don't know.
cisplatin?
And it's
Oh,
cisplatin.
did I just like take you out of
I think
there?
it's cisplatin. No, we're good, we're good. We'll come back. Is it cisplatin? Yes. So this is cisplatin, and the killing with peptide 1 and 2 is about 24 to 35% killing. Cisplatin in the same experiment and the same parameters killed about 40. So we have one of the peptides that killed almost alone, without help. Okay, killed almost close to cisplatin killing. And if you go and look at safety, because this is the most important thing, when you develop something, they will ask you, is it safe or you want to kill us? So it is very safe, up to 40 milligrams per kilogram. Did not affect body weight, spleen weight, liver weight, histology of all kidneys, heart, everything is beautiful, very safe. 40 milligrams per kilogram is a very huge thing, very huge dose. We actually then looked at the most important question: how do you know that your peptides are stable? So we actually sent this to our liquid chromatography mass spec from a small grant from the beautiful Mott Center. Thank you, Dr. Moore. And we came out with this beautiful result. So this is endogenous, and that there is something resembling at the comes dilution at the same time at this minute. 3.33. So this is something endogenous, we already know. This is our standard. We inject the peptide, the standard, and we get dilution time of 2.94. Of course, this is the peak that comes endogenous with it. And when we injected our peptide, we got most of the peptide diluted at the same time, maybe another product, very small amount, which could be a cleavage of 1 or 2 peptides, it could be. I forgot to tell you that when we did design the peptides, we made them the D orientation. Does that make sense? DL. Which one? Which one? So in sugar, D is the natural, L is the mirror image, yeah, enantiomer, right? In amino acids, it's the other way around. So D is the L is the natural, L peptide, and D is the fake one, the other one, enantiomer, yes? So we made them the D orientation. Why? So it protects the enzymes. Aminopeptidases cannot digest the D, they only digest the L isomer. So we made this nice trick where we did the D orientation or isomer. So long story short, 2 hours, which is more than enough to be absorbed through the circulation into the inside. Very safe, no degradation. And the PK is 20 minutes, which is very high. Half-life is 40 minutes. So in 40 minutes, you lose half the dose. So now when we submitted this for funding, the reviewer said, everything is great. The only thing that we don't like is peptide therapy. It's not good. So I want to tell you, you know of a peptide therapy? That's clinically approved, FDA approved, and we use it every day. Lupron, what else? Come on. Ozempic, GLP-1 agonist, come on. Anyone? No? Peptide therapy? A lot, yes. Insulin, that's a big. Good job. But there are many peptide therapies that are FDA approved and used. Okay, if you have good safety and you can inject with a high dose, because the idea is inject with a high dose, so it can get absorbed quickly before it's degraded, and that's the whole idea. And 40 milligrams per kilogram, and we're good. So it's safe, it's stable, it's absorbed. Okay, this part of the study is not done in my lab because I have no clue how to do this. Okay, so this is done in Sweden, University of Sweden Hospital, where they ran 204 samples from 204 patients. Okay, they found 98% of the patients were LMTK3. What do I do? LMTK3 positive.
On the bottom.
Okay, so they found LMTK3 everywhere. 204 samples from stage 1 and 2. They only did stage 1 and 2. Okay, 204 samples. Not almost they're all positive. Okay, now the nuclear part was associated with improved prognosis. Remember my theory? Nuclear, okay, cytoplasmic is the bad one. Okay, now they did something called univariable analysis and multivariable analysis involving many parameters. Okay, so they found that patients with stronger cytoplasmic than nuclear LMTK3 staining, higher risk for death. They're at higher risk for death. 75 of these patients died within 5 years of the diagnosis. Multivariate analysis shows cytoplasmic was not very significant, but also the favorable effect of the nuclear one remained. Now, when they did, you can see cytoplasmic ratio, where is it? Where cytoplasmic is more than nuclear, which is the blue, you see it's very significant, which means more cytoplasmic than nuclear is worse, is bad. The opposite is good and favorable. So now they are running, I think I have the data, I just got them, I didn't have time to put them in. 400 from the phase 3 and 4. They actually found, and I told you, they actually repeated all the experiments that we did, and they all confirmed it. So remember I told you about alpha V and beta 1 that we found with a mass spec, the list, who remember? Last year I told about this, 5 minutes ago. So here we found alpha V beta 1 integrin. They are dimerized together. They are a receptor. They actually, remember when we were looking for CD11B and CD18, the beta 2 integrin as a receptor for myeloperoxidase, it turns out to be, it is alpha V beta 1, and that came out immunoprecipitated with the mass spec data. So alpha V beta 1 actually dimerize and act as a ligand that actually binds to myeloperoxidase. Okay, so we developed this theory or mechanism where LMTK3 actually activates, I didn't show you that part, but it activates the ligand alpha V beta 1, make it active, and this actually inactivate and change MPO from inactive to active monomer MPO that takes nitric oxide to nitrosodium cation that actually inactivate caspase 3 and shut down apoptosis. So this is what we came up with for the proposals. Is this inactive, the dimer, the integrin part? Monomer MPO will not do anything. So this is just to show you the pattern, and this is to show you where we are right now. We're in phase 1 clinical trial. The name of the company, if you want to invest in the stocks, is called Therapeutic. They're funding my lab currently, and thank you for your attention.
Thank you so much. So if this has no side effects on healthy cells, have you thought about using it as a preventive or preventive of reoccurring cancer? Or also maybe prevention for people who are susceptible to cancer?
Have we thought about it? Yes. Did we do it? No. We have a lot to do. But it's good point. Yes,
So this is an interventional clinical trial, so it's past the drug development hurdles, like it's been tested in more advanced mammals, or is this like a biomarker observational study?
one more time.
So is this an interventional clinical trial you're doing, or?
Yes, no, this is just what do you call it? This is just looking at the expression, that's all.
Just looking at the expression,
Just the
okay,
expression
Gotcha.
in patient samples.
okay,
It's not any study yet.
gotcha,
Yet.
gotcha.
So I may have missed this. Do you know how the peptide is taken up by the cell? And how does it specifically touch? So does it only target the kinase in the cytoplasm and not in the nucleus? Does it prevent the,
Good question.
does it
So-
prevent the localization?
So, very good question. So, LNP is expressed in the membrane also. So the peptide actually binds and inactivates the signaling mechanism of that part without the need to go inside you.
So have you looked at whether in cells, does the peptide affect the localization in vivo or in the mouse models?
Does it affect
Have
the
you shown the
localized?
specificity of the peptide in the in vivo models?
The only thing we saw in the cell culture, they co-precipitated them. We didn't
The
see.
reason I'm asking, because I know there's these kinase inhibitors to these, and have you tested them side by side?
No,
So
we did
peptide-
siRNA. We
I'm
knocked
just
down,
thinking for-
we just knocked down the LNPK3 with siRNA and we got the same results.
I'm just saying because I know sometimes when I try to target using our new compounds, they always ask us to do side by side comparison
Sure.
with inhibitors that are already out there. So I'm curious to see how this peptide compares to.
So the good part is that it doesn't go into the nucleus. Because when you treat normal cells, nothing happens. And with even very high dose. But you're right, there are many kinase inhibitors that we can try.
If this one's more specific, then because a lot of kinase inhibitors are dirty, so maybe the peptide is more specific. I was like, it'd be nice to show.
Yes.
So the antibodies have the same effect as the peptides?
Yes.
That's what you made the peptides because you couldn't patent the antibodies.
Correct.
Right, so the antibodies would be your cosmic control then.
The antibody only affect the membrane one.
Right, because it's.
But in case there is no membrane and only cytoplasmic, which is the case that we have seen in stage 1, is they are only mostly expressing it on the cytoplasm, not even out yet.
Oh,
Yeah, we expect to see in the late stage, that it will be in the membrane and out. But it looks like in the first study that we did with the 190 or 204 patients from phase 1, it looks like it's localized to the cytoplasm.
how does it get outside of the cell?
How does it get outside of the cells? It's secreted protein.
It's not attached to the membrane.
It is attached and secreted. All three: nuclear, cytoplasmic, no, actually 4. Nuclear, cytoplasmic, membrane, and released. And do we see a difference between all 4? We don't know yet. We still don't have enough information about the exact mechanism of how this is being works. But it's an accident that led to it.
So the goal is early detection biomarker. This is not an interventional.
It is interventional.
It
If
is
you shut it down, you actually induce apoptosis. And it's as effective as cisplatin. I showed you.
Right, that's why I was asking. So, but there's a big there's a big drug development pipeline before you can even get an IND for a phase 1. So is this going through that process? Like, is this being tested in dogs, monkeys, whatever?
Just 3 animals, 3 different animals.
Gotcha.
It's already been done.
Does it matter the state of the cancer, like level 1, 2, 3, or at what stage would do the best?
So here's my hypothesis. Till now, it is true. And I don't think it is only specific to ovarian cancer. I think it's specific to any inflammatory disease, even fibroids. I would expect to see LMTK3 is produced outside. So Any cells that are going through stress actually start releasing this outside. The degree of stress is what determines where it goes. It looks like where it goes has a different function. That I don't know. I really don't know. Enough questions, guys. Aren't we time up?
Very good. Any more questions? Thank you so much, Dr. Saed. We're going on a break. We have fresh coffee and cookies. Please help yourselves, but please be back at 4 p.m. for the rest of the meetings.
Yes, we will. We will.
Good job.
All right, so we're going to start the last session of the seminar. So it is a pleasure to welcome Dr. Ira Winer. Ira is a physician scientist, gynecologist. He is associate professor in the Department of Oncology and OBGYN here at Wayne State University. The clinical practice at Hermanos. Ira is part of the phase 1 clinical trials program, which conducts clinical research studies on new investigational cancer drugs. He's been a recipient of several awards. The most recent one is the 2023 Wayne State University School of Medicine Teaching Award. And he has been named by our magazine as Detroit's top doctor 5 years ago.
Thanks. Thank you very much for that.
You succeeded embarrassing him.
You did. It doesn't take much. Don't have much hair to hide it anymore. All right. So again, thank you for inviting me to talk. I'm going to try to get to this hopefully in under the 30 minutes. The goal of this talk really is to again, similar to the prior talk, to give a little bit of a journey from basic science through translational, and it's going to end with a current trial that we have open in Carmanis right now that we are actively accruing patients to, based on a lot of the data, some of the data that I present here and some of the data that is from others as well. So disclosures for that investigational trial that we have, there's funding from Chimerix, which was previously a company called Oncoceutics. They're helping with some of the evaluation of the tissue and things like that. And for some of the studies that we did is some funding for Gail Purton Fund, which is a fund here at Wayne State, Jacqueline Bailey Foundation as well.
Silver Family Foundation, and the Michigan Ovarian Cancer Alliance. So as we've heard throughout the day, there are a lot of targeted therapeutics for ovarian cancer. Unfortunately, a lot of those have failed. And the reason for that is that ovarian cancer is not one pathway. It is an interplay of hundreds, if not thousands of pathways. And so when you target one pathway, a lot of times, unfortunately, there's additional changes that occur within the cell that can then abrogate the changes that you made to try to block your pathway of interest. And so I became interested, and I'm going to try to throw some of these in there just so, because it is late in the day, to try to get an idea of, is there a pathway or is there a process that perhaps we can target that actually will target ovarian cancer no matter what pathway of interest you're actually interested in. And so, as we've heard about a little bit before, there's something called the unfolded protein response. And this is a stress response that is present in normal cells, and it's a coordinated cascade of signaling events in response to an overwhelming accumulation of unfolded or misfolded proteins in the lumen of the endoplasmic reticulum. So normal protein do fold, they get secreted or moved to cellular organelles, and they ultimately translocate to the ER by means of a signal peptide. Within the ER lumen, they become glycosylated for the N terminus, and then disulfide bonds occur, and ultimately they help fold the proteins. Now, interestingly, there are multiple pathologic stimuli that can interrupt this normal folding process, and it can lead to accumulation of misfolded proteins in the cytoplasm, which also ultimately lead to misfolded proteins within the ER. And these are just some examples of that. The UPR itself is activated to assist in clearing these misfolded proteins and returning the cell to homeostasis. So this is just a little bit of background. There will be a little picture, so people that are too tired to read this, but I'll go through it. Basically, when we have increase of proteins, there's a sensor, if you will, that's called BIP or GRP78. There are others as well. This is a membrane bound protein that dissociates from the ER membrane, and then ultimately leads to activation of three different arms of the UPR. There's the PERK pathway, ATF6, which we still don't know much about, and IRE1, which downstream is XBP, which we heard a little bit about earlier. So the PERK pathway is actually the pathway that I'm going to focus on a little bit more. But this initially leads to cell survival, but ultimately the interesting thing about the PERK pathway is that with prolonged activation of the PERK pathway, it leads to cell death, and that's related to activation of a protein called CHOP that leads to caspase-dependent apoptosis, typically intrinsic, if you will, so mitochondrial based apoptosis and cell death. IRE1, the main player there is XBP1. It has to be spliced in order for it to be activated. It leads to ERAD, so the ER-associated protein degradation. And again, with this pathway, if you have prolonged activation of this, it ultimately also leads to activation of cell death or apoptosis, but tends to be mitochondrial independent. Oops, that didn't work. So this is just a kind of an overview of this, again, for those of us who are tired at this time of day, but general concept. The upper pathway, the PERK pathway, ultimately leads to cell death in most cases, and the IRE1, in most cases, unless it's very prolonged, leads to adaptation of stress. Interesting thing in tumors is that we kind of ask the question is, well, in tumors, is there a significant upregulation of UPR baseline because of the stress of tumor genesis? There's an increased demand for folding, there's an increase of translation factors, which leads to aberrant protein synthesis. And so there probably is an increase of UPR. But interestingly enough, because of downstream other side effects, in most cells, the tumor, it actually leads to cell growth rather than cell death. But again, going back to the PERK pathway as well as IRE1, if you sustain UPR activation, could you perhaps lead to cell death regardless of chemosensitivity? Meaning we can abrogate that signal and get around it. So again, now we'll get a little bit more into the mechanistic pathway of it. I love this pathway. So how is this translated first? We'll get there in a second. So the first question is it true that in fact UPR is upregulated in ovarian cancer? And again, if you recall, GRP78, which is BIP, is a sensor that is upregulated and activated, and you can see it in the cytoplasm as well as within the ER. In UPR activation, GRP94 is another example of these sensors. And we looked at a TMA, a tumor microarray here, for both BIP and GRP94, as well as others. And we also have done this now in multiple hundreds of patient samples through POA, so through CARIS. And what we found was that, in fact, GRP78 and GRP94 are significantly elevated. And depending on your cutoff, low or high, but the bottom line is that it's in serous, especially, it's upwards of sometimes of 90% of patients. So again, suggesting that in fact, UPR is activated baseline. So what? Who cares? So then we looked at, again, BIP and GRP94 as sensors and said, okay, well, if we look at outcomes, what happens for high or low or positive or negative? And what we found was that the survival of patients with high UPR, interestingly, had better survival than patients with low UPR activation, because there were some, and some of these TMA were low-grade serous cancers also in that, as well as borderline patients. They actually did worse. And we found this both in univariate as well as multivariate analysis. And so the question again is, well, all these patients get chemo, right? So does chemo activate the UPR? Does it lead to prolonged activation? Does it lead to cancer killing from that perspective? So we'll get there in a second. So, and again, this is kind of a summary of a lot of different things, but what we wanted to look at was, are there chemicals, things that are out there that we could repurpose perhaps to use for activation of UPR? And so we basically developed with Dr. Fribley a two-step screen. He had it specifically. Dr. Fribley is a Ph.D. here who does work in ENT, in pediatric ENT. But we developed a specific ovarian cancer screening line for both CHOP as well as XBP. And we found, again, these are three of ones that we found, three agents that actually increase significantly CHOP rather than XBP. Again, XBP being the homeostatic arm, the IRE1 arm. And the three were disulfiram, cholesterol, and ONC201. ONC201 is one I'll get to in a couple seconds. The second part of our screen was, okay, so if it upregulates CHOP, but it doesn't kill the cells, who cares? So not only did it have to upregulate CHOP compared to XBP, but it also had to kill at least 50% of the cells. And this again was on multiplated cells. And so we moved these three drugs just as proof of principle and to perhaps get a little bit more idea of mechanistic concept to ovarian cancer cell lines. And we looked at a lot of different ovarian cancer cell lines. We looked at some endometrioid, some serous, some undifferentiated, some low grade, some clear cell. And we found that pretty much with all these cell lines, both platinum sensitive as well as platinum resistant, the three drugs at reasonable IC50s killed cells. So I'll show you just a little bit. Disulfiram is a very interesting drug. You may have heard about it. It's a drug that is used for alcohol abuse. It's a cheap drug. It's about 50 cents a day. And the interesting thing is, by itself, it definitely killed cells, but also because of what it does, it's a chelator. When you add copper and other different types of magnesium, some other different types of ions to it, it actually impacts and causes even better cell killing. Again, I'm not going to go too much into these, but just know that it did kill cells. We had a nice tumor model, both as xenografts as well as IP model, which demonstrated survival benefit. And it did, in fact, activate CHOP in vitro as well as in vivo. And we actually looked in vivo in the tumors to make sure that the UPR was activated. So again, getting back to the stem cell question, debate, if you will. With disulfiram itself, we looked at this. We did not look at this in some of the others. This was just an interest of ours at the time. What we found was that disulfiram actually activated the UPR within these cells, both in the stem cells, but also in the early progenitor and intermediate progenitor. So it actually activates, and not only does it activate, but it actually kills all three and to the differentiated progenitor as well, which I'm not going to show for time purposes. The other thing that we found was that Taxol, again, going back to our original clinical question was, well, Taxol, right? If we use it all the time for ovarian cancer, does it activate UPR? And in fact, it does. And not only does it activate UPR, but it actually synergizes with all three of those compounds, both in vitro and in vivo. So this ONC201, the reason I'm bringing up ONC201 is because this is actually the basis for our trial. But ONC201, I'll get to the definitive mechanism of action a little bit later, but it actually activates as its baseline UPR, then has multiple downstream effects, including inhibition of the AKT and mTOR pathway, as well as ultimately leading towards cell death via apoptosis. So what you can see here again is just various cell lines that we used. This is a wound heal assay. It's a very easy concept for invasion and migration. We did a bunch of various things again to look and make sure that what we thought we were seeing, we did. We actually inhibited CHOP in this cell line at a later time point along with the company and found that a lot of the effects that we saw were abrogated when we did that. So again, showing that it does seem to be related to CHOP-induced UPR on some level. So cholesterol, we'll come back to in a second. So again, a little bit more mechanistic. How do the cells die? So it does seem, as suggested by UPR in general, and especially CHOP, that it's via the intrinsic pathway. So the intrinsic pathway, again, what we see is you have an increase in apoptosis when you add, this is specifically for disulfiram. We see that with all mitochondrial depolarization. We see a decrease of anti-apoptotic and an increase in pro-apoptotic, again, mitochondrial-based BCL2 family members. And also, what we can see, again, this is by qPCR, but bottom line is that a lot of this is happening via the mitochondrial pathway. Additionally, we looked at CHOP, just again, this is with cholesterol itself, but what we can see is we looked at BIP, again, that's a sensor showing with cholesterol, you can see increase over time. CHOP also goes up. We then got into a little bit more mechanistic analysis, and this was done with Alex Paradon, who was a fellow, and Aisha helped with this as well. And what we found was that there seemed to be an association with XIAP and XAF1, which I'll get to the kind of the whole pathway in a couple minutes. But this was a very interesting thing that we had looked at and found that in fact there may be some connection. So again, caspase 3 and 7, intrinsic pathway, caspase 9, and again, this pathway was of interest, and I'll get to that right now. So, okay, so our next question mechanistically is XAF1 downstream target of CHOP? Again, our hypothesis is that CHOP is the main driver for all of this. And so we did some siRNA experiments. What we found was that when with cholesterol, we had our vehicle CHOP siRNA, CHOP, again, does not go up as it shouldn't, but cholesterol, we know, in fact, with scrambled does increase both CHOP as well as XAF1, but when we used siRNA, CHOP doesn't go up and neither does XAF1. When we did this, looking at our cell lines, again, it's not perfect. Again, none of these are perfect, you know, because they're not, these are not permanent siRNA. These are just, these are temporary siRNA, but what we found was when we used vehicle versus cholesterol in the siRNA, we found, as you can see here, that it did abrogate a lot of the killing by cholesterol itself. So this is a chip assay that we did in the lab. This was done by Marufa Ruman. And we initially looked in the upstream promoter region of XAF1 to see if there were any identifiable CHOP binding sites. And we actually found a number, and the chip actually proved, or somewhat proved, as chip can, that that there is likely binding in this area. And in fact, at least in terms of fold, this is the most active promoter site. So Again, doesn't prove, but again, another piece of information regarding that this XAF1 may be downstream of CHOP. Wish I could say we had more experiments then now, but now we're waiting for funding. So that's what we're working for right now. But nonetheless, we do have at least a picture and a concept. So again, our pathway of interest is more, again, the PERK arm of the UPR. We found that CHOP is upregulated, and if it's upregulated in a significant manner and it's prolonged, it ultimately leads to depolarization of mitochondria, again, induction of pro-apoptotic proteins and decrease of anti-apoptotic proteins, releasing cytochrome C, degrading caspase 9, and ultimately caspase 3 and 7 and apoptosis. It seems that XAF1 is involved in this process, again, likely downstream of CHOP. XIP, again, is an inhibitor of the reduction, if you will, or the degradation of caspase 3. So when we activate XAF1, we get rid of this negative inhibition and leads to apoptosis. Great. So that's again kind of some basic science, mechanistic, translational stuff. But again, translational is the most important aspect. So this ultimately, a lot of this data, as well as some other data from things that I just don't have time to show, led to a current ongoing study, which is the ONC201 with weekly paclitaxel in patients with platinum-resistant or refractory ovarian cancer. So again, going back, ONC201 is an oral drug. It's a DRD2 antagonist, but ultimately it activates the UPR, leading to downstream target inhibitions of AKT and TRAIL and some other things. This has already gone through phase 1 trials and already established a phase 2 dose before we start, actually right when we were starting. So it's 625 milligrams. This has a half-life of about 11 hours. And pharmacodynamics, which means what happens in the cells, not just how the cells respond, but what happens in the body. In the phase 1 studies that we were part of as well, demonstrated that those effects go out to 96 hours or more when you actually dose the drug. So again, let's go back to Taxol. So we saw in our initial studies that Taxol upregulated UPR by itself. It was synergistic with ONC201, again, something I didn't show. But here is some of the data. But the Taxol in and of itself, especially weekly Taxol, has a reasonable efficacy in platinum-resistant disease. It's not great, but it's pretty good. It's about 20 to 25% ORR is an objective response, meaning we see tumor shrinkage. And progression-free survival, meaning to the next type of tumor treatment of 7 months. And then an overall survival of 15.5 months. Now, this was in the largest phase 3 trial. There's a huge caveat here, is that they called these patients heavily pretreated, which were 2 to 3 prior, which is not very heavily pretreated at all. If we look at our patients now are on 4 to 7 lines. So our trial objective, the primary objective, as with most interventional trials, is objective response. What that means is there, again, a measurable reduction in tumor based on imaging? This has to at least be a partial response, so we need to see a 30% reduction in identified tumor. But we're also looking for something called clinical benefit rate. What that means is, can we keep patients on this for a prolonged period? They tolerate it, and they even have stable disease. And that also includes partial responses too. But again, the goal is to get to 6 cycles of Taxol and then continue on the pill by itself, and it's a weekly pill. So it's something that people tolerate pretty well. Duration of response is something else we look at in trials. This is if a patient has a response, how long does it last? So if someone has a response and it shrinks by 50%, but it grows back within a month, does that matter? The answer is probably not, unfortunately. Another endpoint as a primary efficacy point was progression-free survival. And again, that means the time from the initiation of the treatment to their next progression, meaning their next type of treatment. And we've got a lot of exploratory evaluations that we're using. Again, we actually have tissue biopsies that we're looking at, making sure that we see UPR evaluation and activation. Pharmacokinetics is how the drugs are cleared from the body. And we're making sure that even though it doesn't appear from other trials that Taxol and ONC201 interact, if it somehow changes it, that could. So it's important to know. And there's also some newer data that came out right before we started this, looking at the immune system. And there may be, again, in UPR, there's definitely some immune changes that we see. And if we can activate the immune system as well with UPR, ultimately that might also be a mechanism where we attack the cancer. So this is the trial schema. This is what we did. So this was actually a phase 1B. What that means is we wanted to make sure it was safe. So we had a certain number of patients that we had initially. We actually didn't have to dose reduce, meaning reduce the dose of either the Taxol or the ONC201 at all. So we had 6 patients and deemed safe. So we moved on to part 2. And currently we're in the process of accruing patients to this. Right now we have 19 patients that have been accrued to the study so far. We want to get to 34 before we make our interim analysis. So we'll look and make sure it's efficacious. There actually is some statistical data that we're actually going to look at again, given the fact that some of these other patients are quite heavily pretreated, to see kind of if our numbers can be a little lower, because most of the patients before, again, had 2 or 3. I'll show you our numbers in a second. And all of those patients were not platinum refractory in the prior trials. So these are very preliminary results. So again, we have 19 patients. That's the 19th patient is not included because she's still in her about to start her 3rd cycle. But we have about a quarter are minority patients, which include African American and Asian American. The majority currently are Caucasian, but again, we have a small number of patients. The age is as noted. This is the most important thing. The mean number of priors, we're allowing 7 priors. And the mean number of priors, meaning the average, if you will, is 5 for these patients. So they're quite heavily pretreated. Now, I would call these patients heavily pretreated as compared to some others. We also have 4 platinum refractory patients currently who have been enrolled on this trial. So to date, we've seen 2 patients with a partial response. That's 12%. Again, and heavily pretreated. That's actually not a horrible response rate. The clinical benefit rate for these patients currently to date is about 40%. So it's 7 out of 18. We've had 5 patients with stable disease, as well as the 2 partial responders. The duration of response, the mean average is about 5 months. So it gets out to a reasonable amount. And the range, actually, with 1 patient that was out to 442 days, she was on the pill by itself without any other chemotherapy. Ultimately, she progressed, but she was doing great until that time. So that's kind of where we're at right now. That's the data that we have. But it shows, the concept of trying to move something not just from the lab to the lab, but trying to move stuff from the lab to translational. And the collaborative nature that we can do that with both as physicians, as clinician scientists, as PhDs. But that's the end goal, is to try to get these to patients so that we can hopefully make a change and make hopefully a positive change in these patients. So there's a lot of future directions that we're working on, but the bottom line is that's where we're at this point. And this on the left was really, these are from the prior studies, but really for the trial, it's obviously the patients and the families and our clinical teams, and that includes my partners. Of course, Dr. Morse is already on here, but Dr. Walbillick and Dr. Gogoi. And hopefully soon we're moving this up to Flint to try to accrue more patients there, and hopefully a more of a mixed patient population as well. So that's where we are. And thank you guys for listening.
Wonderful talk, I would say. So with the GRP78 data in the clinical trial, did you collect any serum?
Yes.
GRP78, can you look at antibodies for it?
So there is some data now that actually it gets secreted like a lot of the other things we were talking about earlier.
To do IHC.
Correct. So we do have serum, and so we're going to look. We only have a small percentage of our patients are going to be in that kind of that PK/PD, so the pharmacodynamics.
Yeah.
So we're going to have about 10 patients, 8 to 10 patients. So we're going to look both at serum as well as IHC and see if we can pick it up, because again, this is a biomarker, as with a lot of the other ones, that get excreted. So yes, we are looking. And GRP94 also gets excreted. So, but
I know, from our data, because we've also looked at, so actually, I don't know if you're on my talk, but Rita here has some data on that PP2A activator, which actually is a derivative of a neuroleptic, actually. It also induces PERK so maybe we could talk offline.
Yeah.
But so we also see the same thing with the GRP78. That's what I was asking, and we see it induced in platinum-resistant models. So I'm really excited that you're doing these studies, and I would love to continue and maybe test our activator.
Yeah, we can definitely talk about it.
Right, add to that, thank you very much. It can be detected in the blood, so there is a nice ELISA if Connor is looking. But there is another role also, which we need to discuss related to the immunological aspect. This is a very strong anti-inflammatory. So this is just wondering if the changes that may be happening also with the treatment and so on can modify the inflammatory.
The answer is 100%. We know that, yeah, 100%. And there's been some preclinical data that's looked at that, but it's not been translated yet.
Yeah.
There's also some data with PARP inhibitors in UPR, and so
let me add to the.
I put all three of those together, and you got it.
The picture that you show, that middle pathways, I agree with you 200%, and especially the recorded very heavy patients, loaded patients, that tumor has evolved. So it's not that it has 2 million pathways; it has now 4 million pathways. Right. How we can prevent that we reach that stage of the 2 million pathways? Is this approach is not maybe better?
So the answer is ultimately is you want to use these approaches early.
Correct.
Similar to why, we found actually even with PARP inhibitors, we thought in the late stage it was actually effective, and we're finding now that in fact it may actually be detrimental. So the answer is to move it up front. We need some data first to prove that it's efficacious. It's becoming a little easier now in the phase 1 world, in the phase 2 world, to move things earlier. But FDA won't really even look at some of these until you show some efficacy first. So that's the problem.
Ira, had all of these patients been on Taxol twice before? At least once, I know, but twice.
So they had never. So none of the patients had seen weekly Taxol.
Okay.
They had seen, a lot of them had seen Taxol more than once though, but they had not seen weekly Taxol. There were a couple, I shouldn't take that, I take it back. There were a couple that had seen weekly Taxol. And interestingly, when you add ONC201 to it, they actually did better.
So I think that was where we're going. Yeah. I think that's a really interesting.
And it's also interesting, we know kind of our baseline for recurrent and platinum is going to be Taxol Avastin with weekly. And that's that's still the standard. The problem is a lot of patients can't get Avastin for many different reasons. And so this drug or others, might be a replacement for that. So.
Ira, did you talk about the toxicity of the combination?
So we've had so far to date, so we did not have any dose-limiting toxicities in the 1B. We've had one patient, and again, all these were heavily pretreated. One patient and we're starting at 80 for weekly, or 3 weeks on, 1 week off. We've had one patient that had in her 3rd week, we had to hold her because she had an ANC that was 0.7, 0.6, but that was the 3rd week. And so she was dose reduced to 60 and has done fine since that time. So there's been actually limited toxicity. ONC201 for the first 24 hours can cause a little bit of dizziness. So we recommend people don't drive the first 24 hours. So they have to have someone bring them, because what we're doing is you take the pill the day before based on the pharmacokinetics, and you get your Taxol the next day. So there are some, but people have tolerated it relatively well. ONC201 has also been used in glioblastoma. It's been studied in uterine serous and failed miserably, unfortunately. But there is something, they had no responses. But again, it's a different disease. So
potentially you could add that to it. Right?
You could 100%.
Research, when you do a treatment for a cancer, and then it comes back, Do you compare the microenvironment before and after, and then
Great question. So the answer is, so there have been limited studies looking at that. We're actually looking at a study right now. Again, this is through an alliance where we're actually looking at patients who have had serial biopsies, not only looking at NGS, because that probably, it changes a little bit based on some preliminary data, but the microenvironment is the likely thing that changes the most. And so we've been looking at that. We have now, I think we have a series of 50 or 60 patients. We're just starting to look at this. So I don't have the answer, but the likelihood is that's what we think does happen.
And if I can add to this, you are right, because we did it with a neoadjuvant chemotherapy pre and post, and the microenvironment changed the responders and non-responders. The interesting thing is that the responders is associated with immunological activation, while the non-responders, there is no immunological activation.
So it was one of the patient with that really, it's like an extreme outlier duration of response. Is there any thought to doing an extreme outlier analysis? Like what's different about that patient?
Not yet, because there have actually been a few that have actually done okay. So the stable disease patients. She was one of them. We have her tissue. So eventually we will look at that. But we kind of have to first get to that 34 patient mark before we kind of make any conclusions. But again, I think the caveat's going to be with the analysis is that if we even see an objective response of 15 to 20% in patients that have had 5 or 6 priors, it's pretty good, because you don't see that with weekly Taxol by itself. So, well, thank you guys.
Thank you. Thank you so much. So this is a nice part of it. So we combine both the basic science and the clinical and translational. You know, I have been doing research in this field for, I'm not going to tell you how many years. You start calculating how old I am. But we always talk about how the tumor behaves, how the animal models, and the majority of us, we always acknowledge that we are doing this for the patients. I tell you, many years ago, I was giving a talk also, and one patient came and I said, very nice, thank you for all what you're doing, but did you ever ask us, how do we feel? How do we see your research? Did you ever ask us if we understand all what you're doing? And I promised that patient that anytime that we organize a meeting or a symposium or we talk about a cancer, we always will have a patient, a person who is living what we are trying to solve. But not only that we send a message to the patient, hey, we're working for you. We wanted to know how we can improve the way that we're working for them. And this, and again, I kept, and I have my witness, I kept that promise for many years, Yale and here in this first symposium. And it's a pleasure for me to introduce our last speaker, the most important speaker of today, that is Mary Groll-Brown. Mary is also a history and knowledge in reproductive biology and female health. He is a reproductive biologist and female health education specialist. She worked both in healthcare and research for over several years. We're not going to say how many years. We are in the same team. We are in the same team. She works in the Michigan Medicine and Department of Radiation Oncology. But more important, she is an 8.5 years ovarian cancer survivor. Many more. We're counting on you. So we are delighted that you're here and to share your experience. And I'm sure we're going to learn a lot from you.
Oh, thank you so much. It's so kind. So how shall we start this up? Because I have to have my cheat sheet.
You can actually like sit here at the head of the
Okay. At the helm here. Okay, well, thank you everybody for having me. It's just such a privilege to be anywhere except in an infusion chair or a hospital bed. I'm just delighted to be out of my hospital cage. So it's wonderful to be here. Well, as mentioned, I am Mary Groll-Brown. I am an 8.5-year ovarian cancer survivor. Of course, before I begin my discussion, I would really like to take this opportunity to thank all of you that have made it your life's work to eradicate this disease in such a variety of capacities, from scientific breakthroughs and discoveries to the development of pharmaceuticals, administration of the therapeutics, and the delivery of care during the entire journey. Without your ingenuity and expertise, these survivors would be non-existent. I think it's also very important to thank all of the research subjects who came before me for through their brave participation in clinical trials. New therapeutics have become available to patients like myself. Now it is my turn again to be participating in clinical trials in hopes of finding yet another treatment breakthrough, and of course, with the major aim of eradicating this disease. A little bit about myself. I was diagnosed with stage 2B ovarian cancer in March of 2015. As of yesterday, I started my 15th line of defense. I have had 2 abdominal debulkings, 12 different oncolytic regimens, and I received palliative radiation therapy. 3 of these treatment modalities have been clinical trials, 2 which were and are phase 1 trials. I myself have worked in healthcare since I was in high school and in various departments and clinical settings. In my late 30s, I decided I wanted to work in clinical research. I went back to school and obtained degrees in neuroscience with an emphasis in sleep medicine, biomedical science with a concentration in reproductive biology, and then under the umbrella of public health, a degree in healthcare education focusing on female reproductive health. It was after obtaining my last degree that I was hired at Michigan Medicine. Before going out on medical leave a few years ago, I spent the better part of my career working in maternal fetal medicine, co-addressing sleep disordered breathing with maternal fetal outcomes in high-risk pregnancy settings, as well as in reproductive endocrinology and fertility patients. After my initial cancer treatment, when I did have a return to work, I decided to switch gears and work in cancer research, as I could bring patient perspective to trial design, execution, and delivery of care. Although there is really no way to prepare oneself for cancer diagnosis and battle, I do accredit much of my background to have been able to navigate this rocky road and to tease out some ancillary services that have kept me able to endure all that this disease, the treatments, and the sequelae bring about through the cancer trajectory. Being asked to speak about survivorship today had me realizing how different this might look patient to patient, recalling what I witnessed with my own patient base while working in radiation oncology. We all understand that survivorship is the time frame from which one is diagnosed through the end of their life. It acknowledges that cancer treatment and its multitude of consequences do not end when the specific therapy is completed. The multitude of transitions one goes through across the phases of survivorship are often very imperfect. This supports the need for a defined plan of care for survivorship, not only to facilitate these transitions, but with patients living longer and more therapeutics becoming available. We can only imagine what this need will look like clinically in the near future, if not already. Sadly, there is no standard of care for survivorship, and not all cancer centers offer the same comprehensive services. Of course, the oncologist is very well prepared to manage symptoms such as surgical site infections, low blood counts, chemotherapy-induced nausea and vomiting, sexual dysfunction, and a myriad of other adverse reactions. The primary care physician can also be instrumental in managing some side effects one will experience and can be very supportive throughout survivorship, especially if you have had a long-standing relationship with your provider. But a subspecialty that has enhanced my survivorship is physical medicine and rehabilitation oncology. Intuitively, we think of PM&R services and orthopedics, sports medicine, stroke clinics, and for amputees. But how logical is it to have a physical medicine service as part of the survivorship care plan? Cancer can easily be defined as a severe and chronic disease. Impairment and debilitation will most definitely become a piece of the cascade of events that takes place throughout one's survivorship. However, patients can maintain much of their functionality and independence with the proper identification of issues and implementation of treatments cancer-related dysfunctions. PM&R oncology physicians or physiatrists are providers that have special training in cancer-related neuromusculoskeletal and cognitive impairments and debilities. This service also encompasses physical medicine and rehabilitation, neuropsychology, oncology, and palliative care. Cancer and its treatment often cause physical, mobility, psychological, and cognitive problems. These problems can make it harder to do daily activities, return to work, or even continue with cancer treatments. They can have a lasting effect on your health and overall quality of life. Cancer rehabilitation can help manage these problems. Ideally, this service should be established when establishing care with the oncologist. Rehabilitation is the actual first stage of PM&R care. This is a preventative treatment plan that is developed after the diagnosis and before the commencement of acute treatment. The second stage is restorative treatment. This stage begins while the patient is on and/or following treatment after surgery or other treatment modalities as well. This intervention attempts to return patients to their previous levels of physical, psychological, social, and vocational functioning. The third stage is supportive treatment. This involves patients whereas their cancer has become a chronic health condition. Interventions are now inclusive of how to accommodate fixed disability and how to increase self-care ability and minimize further decline from ongoing disease. The final stage is palliative treatment. In this stage, care is centered on minimizing or eliminating complications and providing comfort and support during the terminal stages of the disease. There is most definitely overlap with multidisciplinary teams, but the concentration of physical medicine and rehabilitation oncologists is preservation and restoration of functionality, maximizing independence, and improving the patient's quality of life. The interdisciplinary team is inclusive of, but not limited to, physiatrists, neuropsychologists, physical and occupational therapists, speech pathologists, nutritionists, and others. Within the state of Michigan, there are only a few facilities that offer rehabilitation oncology service. Beaumont Cancer Center, Mary Free Bed Rehabilitation Hospitals in Grand Rapids, Muskegon, Saginaw, Traverse City, and possibly at some of their other sites, and at Michigan Medicine's Rockville Cancer Center. It was very early on following my initial surgical procedure and chemotherapy treatment that I recognized some concerning health changes. When I was discussing with my GYN oncology mid-level during one of my 3-month follow-ups, I was told about PM&R oncology, a fairly new service at the time at Michigan Medicine. This was approximately 9 months after my initial diagnosis. The first issue that was addressed for me was unilateral lower extremity lymphedema. I did have 17 lymph nodes excised from my right groin, so when I became symptomatic, PM&R oncologist triaged and referred me to our lymphedema clinic. I worked with a specialized occupational therapist for a long while and was able to not only relieve the pain and swelling, but prevent progression through lymphatic massage, compression, exercise, and aquatherapy. Shortly after this incident, I noticed that I was becoming symptomatic of sleep disordered breathing. PM&R put in an order for a sleep study, and I couldn't believe that I had developed severe central sleep apnea with 36 events per hour. Having worked in sleep medicine for a number of years, I knew that my baseline polysomnography score was 0 apnea events per hour before becoming ill. No one really understands the mechanism that caused this change in my brain, but I use CPAP therapy every night to keep my apnea within normal ranges. From this point on, the more therapies I underwent, the more health issues I developed. Of course, as I ended certain treatments, some of the problems would be moved over to the past history list. But besides impaired sleep and lymphedema, some of the more prevalent health issues that I have had addressed through PM&R oncology and neuropsychology entail physical difficulties, including decline in balance, difficulty walking, poor endurance, muscle spasms, muscle weakness, and cancer-related fatigue. Emotional effects of cancer, including stress and anxiety, post-traumatic stress disorder, and depression. Uncomfortable sensations, including bone pain and acute episodes of pain crises, myalgias, arthralgias, joint stiffness and reduced range of motion, as well as numbness in my hands and feet or peripheral neuropathy. I experienced bowel and bladder issues, radiation fibrosis, and radiation enteritis, edema, scar tissue restriction following surgery, swallowing difficulties, and poor appetite, weight gain, and at times weight loss, osteoporosis, and bone fractures. I have not had any cognitive difficulties such as with memory, concentration, or attention, and I have not had voice or speech challenges. Another huge area that rehabilitation oncology assists patients with is navigating both short and long-term medical leave, FMLA paperwork for employers, Social Security disability form, and the determination if the patient will be deemed permanently disabled. This aspect of survivorship can be a stressful and very confusing piece to the cancer care spectrum, yet so vital when the patient is fiscally dependent on these benefits. As treatment modalities evolve, so too must the research on short and long-term side effects and multi-system management over the continuum of care. Rehabilitation services are elements of standards of care for many neurological, cardiovascular, and orthopedic diagnoses, but currently are not standard for actual or potential dysfunctions among cancer survivors. Both the disease process and the variety of therapeutic modalities increase risks for dysfunction, impairments, poor survival, and diminished quality of life. Rehabilitation focuses on optimizing quality of life and maximizing function throughout survivorship. Healthcare professionals should be urged to integrate high-quality interdisciplinary care to promote collaboration and dissemination of knowledge, which will yield better care for cancer survivors. In conclusion and recap of valid points, the cancer survivorship journey begins at the time of diagnosis and continues to the end of life. Needs of cancer survivors are often complex and change over time. Cancer rehabilitation programs that prospectively identify and address these needs throughout the cancer journey can provide an important component of high-quality survivorship care. Adding this additional layer of oncology care can optimize the health and well-being of survivors, effectively manage new and existing symptoms, prevent development of late effects, and promote good health behaviors. These aims can transcend wellness and a higher quality of life compared to those that are left untreated. I know that this multidisciplinary approach in my care continuum has kept me as well equipped mentally and physically as possible to continue battling my way through this chronic disease. It has strengthened my will to live through easing mental and physical sufferings and has certainly enhanced my survivorship over the last 8.5 years. And I thank you for allowing me to share that with all of you.
Thank you so much.
You're very welcome.
And I don't know if anybody has a comment or anything that you would like to add.
Yes.
My neighbor has stage 4 brain cancer. She started therapy yesterday. So I thank you so much for sharing your experience, because I'm gonna speak with her about some of the.
Oh, wonderful. Yes, it was really something in other departments that I've worked in. They're right there. I worked in neurology, I worked in orthopedics, and of course, it's just part of the team front and center. And I just even working in the field, I had no idea what I was up against. I was a really healthy person at the time, and diagnosis and what a cascade of events. It's like you're just always tumbling downhill and clawing back up a little bit and then getting knocked right back down. And I've had lots and lots of bad news and heavy side effects and near-death experiences. And a year ago, last March, was given 1 month, and I'm still here. And much of that is attributed to PM&R getting me up out of the hospital bed right after a surgery or while I'm inpatient and keeping me moving. My neuropsychologist is here today from Michigan Medicine. Nicolette, raise your hand. She's been keeping my monkey mind in place and in check for a number of years. I've had 4 different psychologists through this journey, but I've had a lot of new and very heavy mental pieces that come on board going through traumatic experiences, and the skills and the tools that one obtains through these services is, I don't know how people get through it without it. And I just have tried to do a bit of advocacy through the years just to kind of share these experiences, because when we are working in direct patient care and we have patients before us, they're not very candid about what they're experiencing. And I think we need to do a better job of teasing that out. Dr. Winer and I were talking about that this morning, since I'm now in his care. I got booted from U of M because they ran out of treatment options for me. So I'm a newbie here at Hermanos, and we were just having that conversation this morning. It's really hard to maybe more metrics when patients are doing intake to tease out some of these issues that patients are having. Like, I worked in radiation oncology, and I was not prepared for anything that I got hit with when I went through my radiation treatment. And I thought I was crazy. I was like, my patients didn't have all these issues. And when I did a little rudimentary background research on it, it's in the literature supports that patients do not want to discuss sexual dysfunction. They do not want to discuss enteritis. They do not want to discuss some of the embarrassing, bowel incontinence, or, bladder incontinence, or even pain. And I think psychological pieces, people do not want to admit they need psychological help, or maybe they don't even know that they do need it or that there is help. So I think some of the point of my conversation with this, and I worked in my department trying to make a breakthrough with that. How do we get patients to be with these short times in clinic? Who can tease this information out of these patients as to, address these things? Because yeah, we're eradicating the disease, super, but if you can't get off a chair, you can't get off your toilet, you can't leave your house, what kind of life is that? And so I'm kind of getting to that stage where I have so many health issues. And then with COVID, that was a big horrible piece. The isolation will wither you on the vine faster than anything. So that's a big piece of my rehabilitation, is to get back into the world. How do you do that? So rehabilitative medicine is a good step forward, but it doesn't get a lot of credence for some reason. It doesn't catch on, and I think U of M's doing a very good job with quite a robust program. And I wish for all patients they could have this opportunity.
So the majority of us are here affiliated with Hermanus, and notably absent from your list was Hermanus Cancer Center and the DMC's Rehabilitation-associated Michigan, which is pretty robust. So do you have any insight into like the program, like how to establish such a program? I mean, you made a very convincing argument for it, but I guess like, is it?
Yes, of course.
So we do have a really good supportive, like we have a robust supportive multidisciplinary team here, but we don't really have that we can talk about. So do you have any insight into how to build such a program or start?
So yeah, like a start, even with any kind of physical medicine and rehabilitation service in the hospital at large, I would think, I don't know if you have that residency program here or the fellowships. I'm not sure what Wayne State has exactly, but a great start is just reaching out to, obviously you're going to have mobility issues. That's one of the biggest things with most therapeutics, especially your legs, walking, pain. So that right off the bat is certainly under the umbrella of physical medicine and rehabilitation, which most hospitals have a center for that somewhere. So a phone call into that, or welcome residents that are going through the rehabilitative medicine with the concentration in oncology, because they're out there. There are a few programs, and Dr. Sean Smith at U of M is the division director, and he has a font of information and resources. He now has another associate. They started out going to tumor board and integrating themselves into the oncology, center at large. He sees, several disease sites, but he was having clinic days in GYN oncology. He now has an office space not only in the PM&R building, but he's also over in the cancer center with clinic days there for cancer patients. And it seems you can tap into that department and go from there. But I would think that it would be with Nicolette.
Oh, it's just gonna add in that it's one of the newest subfields in physical medicine. So there are very few fellowship level training opportunities for MDs, but that's growing over time. So we have one at Michigan Medicine where we're training fellows, and then they go out and start their own programs. So it's very possible that it'll be coming to you. It's just kind of right on the cutting edge. And I think that has a lot to say about the change in survivorship and how long people live now and all of the downstream effects that come from that.
And with the new oncolytics, there's a whole new set of symptoms. And oncologists aren't, how can we know if it's a new drug? How do you know what to expect or warn your patient of? So it's important to have those metrics. Like I don't know what the questionnaires are at this time, because I've been out of the workforce for a while, but say when I worked in orthopedics, like the PROMIS, metric. And then if a patient scores a certain way, I think there's algorithms that are, in the system that would automatically kick a patient to a referral. So even if you don't have that actual oncology PM&R physician on hand, there's a PM&R physician somewhere that can be of great use for patients to recover.
you know, the educational aspect, what you mentioned is so important, and this education should start very early. I remember when you introduced the field with the medical students will rotate through OB and Gynonco, the lecture from survivors. And after several years, the feedback that we got that was the best lecture they ever had during the rotations.
Oh,
Yeah.
wow.
So I hope that we can introduce also here. And I really, I don't know if there is any question or any comments before anything else? If no, I know we are getting late, so I want to thank you again
Well, thank you
for
so
coming
much.
and sharing your experience with us. It's the best part of our meetings.
Well, thank you so
And,
much. It's
and we
very
want that this will continue educating not only the people who are here, but also the medical students, the nurses and everybody.
Yeah.
That is our task,
Oh, that's
and we
lovely.
will work on that.
Wonderful.
And again, thank you so much.
Thank you so
I
much.
will- So again, I want to thank everybody who participated today. We will be wrapping up the program, the first part of the program. For the speakers and for anybody who is part of the panel, you're invited to stay. We will open a Zoom exactly what happened with Mary. We want, there is many Marys outside, trust me. We want to know more, but in their own words, of what you are doing. So we will open that panel maybe at 6 p.m. Oh, we're 5 o'clock, so we're okay. So we open the panel at 5 o'clock. Yes, 6 o'clock, sorry, 6 o'clock. So there is a time to rest. For the speakers and the members of the panel, there is some food, so we feed the body as well as the soul. And for the rest, thank you so much for coming. Please share, send your messages that there is a 6 o'clock. This talking to the experts
On the same.
for the public,
On the same.
and it will be in the same Zoom.
Oh.
Yeah, will be in the same Zoom, so you don't need to change the Zoom link. And again, thank you to the organizer. Thank you to everybody to contribute to making this a successful meeting. And I would like to say, see you next year. Yeah. Thank you.